Advisory Committee, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, its products, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of your travel, lodgings, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Prabha.
Good afternoon, everyone. This is Dr. Prabha Traya. I'm going to conduct the open public hearing session, and I will read your name in the order, and then when I call your name, please unmute your phone and start speaking, and you have 3 minutes to complete your remarks. Thank you. The first name is Dr. Kermit Kubick.
To the vaccine advice. Hello, these are my comments to the Vaccine Advisory Committee on the BNT162b2 coronavirus vaccine. Cover efficacy, structured benefit risk, and follow-up. I have no conflicts except for a lot of elderly relatives. Next slide. The Pfizer vaccine appears efficacious based on 95% relative reduction in infections and T-cell responses comparable to human convalescent sera. See Figure 2, page 22, and Figure 4, page 24. Some uncertainty is introduced by the possibility of mild asymptomatic infection, including among placebo patients, including feelings of pain or injection site effects that may have caused more vaccinated participants to be tested than placebo recipients. Next slide. Benefit risk. I have reviewed the available evidence, and as a Caltech graduate, despite the uncertainties, the structured benefit risk of Pfizer BNT162b2 is very positive. The coronavirus, as we have heard, is a serious medical condition with 3,000 daily deaths. While there are therapeutics, there are no prophylactics against COVID-19. Even in the event of approval of the Pfizer vaccine, limited supplies will require EUA of other efficacious vaccines when available, such as Moderna or others. Benefits of the vaccine include prevention of COVID-19, after the second dose, prevention of severe COVID-19, and preventing COVID-19 after the first dose. Risks appear to be mild injection site pain, fever, or fatigue. Increased reactogenicity for recipients over 55 is more than offset by the benefits of preventing COVID-19 cases in older adults with weaker immune systems. I note that allergy-susceptible persons should be informed of any issues in the EUA fact sheet following approval. The net benefit is highly positive. I would recommend the vaccine to my siblings over 80 and my relative in an assisted living facility where there have been 3 cases for staff and 3 cases among residents. There is a need for follow-up of vaccination effects, including how long after immunization patients are protected, whether the vaccine prevents infections which cause further transmission, and how any violations of cold chain supply, dilution, shaking, or administration affect efficacy. Thank you, Dr. Fink, Dr. Gruber, Secretary Azar, Dr. Hahn, and Dr. Messonnier. They need this vaccine yesterday.
Okay, great. Thank you. The next speaker is Diana Zuckerman.
Yes, hi, can you hear me?
Yes.
I'm Dr. Diana Thank you. I'm Dr. Diana Zuckerman, President of the National Center for Health Research. Next slide. Our center scrutinizes the safety and effectiveness of medical products, and we don't accept funding from companies that make those products. My expertise is based on my postdoctoral training in epidemiology and as a faculty member and researcher at Vassar, Yale, and Harvard, and also previously a fellow in bioethics at Penn. I've also previously worked at HHS and the U.S. Congress. Next slide. Today I'm going to focus on two major concerns on how we can improve the data that are already available. Number one, the 2-month median follow-up is just too short to have long-term safety information and long-term efficacy information, so it's essential that the randomized controlled trial be continued. Number two, there's a lack of diversity in COVID cases. There were 0 Black cases in the vaccine group and only 7 Black cases in the placebo group, and there were 0 cases that were ages 75 and up in the vaccine group and 5 in the placebo group. And it was very disturbing. Wall Street Journal did a chart saying that it was 100% effective for Black patients, for example. So that's one of the reasons why we need more people so we have reasonable data. Next slide. There are also too few severe cases to draw any conclusions. There were only 4 severe cases after the second dose. That's just too few to conclude anything. Next slide. Long-term patients were not, long-term care patients were not in the study. There were about 800 people ages 75 and over in the study, but only 5 were cases. So we want to save their lives, but how can we ensure informed consent to nursing home patients when we have no data to provide? And how many frail elderly or their family members can make an informed decision based on so little information? Next slide. We need long-term data to fully understand if the benefits outweigh the risks for frail patients and for all races and ethnicities and for all patients. And that's why continuing the randomized controlled trial is so important. Next slide. In conclusion, the EUA is not approval, and it should have more restrictions than you'd have for approval. So FDA should require continuation of the RCT while targeting EUA to priority populations, especially healthcare workers. I agree, don't let anyone in the placebo group jump the queue. Second, EUA should not allow off-label use for non-priority groups, whether they're celebrities or anybody else. Off-label use could potentially occur under FDA's expanded access program. And last, FDA should delay access to vaccines by the placebo group unless they are in the priority populations. And I am concerned that the blinded crossover would be would be informative if the vaccine is not long-term efficacious. But if it was efficacious long-term, we would lose that information in a blinded crossover. Thank you very much for the opportunity to speak today.
Great. Next speaker is Peter Doshi.
Hi, this is Peter Doshi. Hopefully you can hear me. Thanks for the chance to speak. For identification purposes, I'm on the faculty at the University of Maryland and a medical journal editor with the BMJ. I have no relevant conflicts of interest and no one's paid for my attendance. Slide 2, please. My experience has been that careful review of a large trial takes considerable time and effort. As FDA has already reviewed the data, I'd like to know whether FDA is confident in the data collection for the primary endpoint, specifically that any unofficial unblinding did not affect the results, and that fever and pain medications did not mask symptoms, thus preventing case detection. I didn't find answers to these questions in the FDA's briefing documents. Slide 3. The dramatic difference in rate of side effects between vaccine and placebo raises questions about how well these trials could be observer blinded. With a subjective endpoint like symptomatic COVID, blinding is important, but it seems fair to think that people could make reasonable guesses as to which group they were in. Slide 4, please. In the real world, the mantra has been to test, 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 but this wasn't the case in the trial. The study protocol says in the 7 days after vaccination, do not test unless, in the investigator's opinion, the clinical picture suggests COVID rather than vaccine side effects. This basically amounts to asking investigators to make guesses as to which intervention group patients were in. My question is, was this kind of judgment ever applied in the days it could affect the primary endpoint? And now I'd like to skip to slide 6 in the interest of time. Possible unblinding would matter less if the trials had been designed to directly test the vaccine's ability to reduce deaths, ICU use, and hospitalizations, as most people assumed the trials were set up to do. So it's great when the data look encouraging, but trials should be directly testing the endpoints that matter. Then there's the duration of protection issue. A vaccine that delivers a 95% relative risk reduction of COVID 2 to 3 months after vaccination is one thing. But for the many people who lack natural immunity and don't get exposed to the virus soon after vaccination, protection needs to last much longer. After 6 months or a year, will the vaccine still meet the FDA's 50% effective requirement? The trials just don't have sufficient data to say. Keeping the trials going with placebo-controlled follow-up will help answer the many crucial questions that remain. For those that do not wish to wait for clear evidence that benefits outweigh risk, an expanded access program can be set up. Access doesn't require authorization. And I just want to end by saying that whatever FDA ultimately does, the full trial data must be made publicly available. Thanks for offering me the time to speak.
Okay, the next speaker is Rabih Hassad.
Thank you. Thank you. I am Rabih Hassad, a professor at Mercy College with expertise in epidemiology, statistics, and mental health. I hereby declare no known or potential conflict of interest. Next slide. This vaccine is reported to have an impressive overall efficacy rate of 95%, consistent across key demographics. However, the primary efficacy endpoint of the trial was symptomatic COVID-19 infection. Participants who may have developed asymptomatic infection were not identified in this trial. Therefore, if as expected, an emergency use authorization is issued, the spectrum of COVID-19 infection that this vaccine is efficacious against must be emphasized. Next slide. Adequate statistical power is necessary for differentiating between an actual vaccine effect and one that occurred by chance. The power of this trial was calculated at 90% to detect an overall effect for vaccine efficacy and may not allow for firm conclusions regarding subgroup differences. Next slide. As reported, there were no serious safety concerns, which is quite reassuring. However, there were frequent mild and moderate adverse effects, which are recognized as common side effects of vaccines. Next slide. Additionally, participants were blind as to whether they received the vaccine or placebo. But given the common awareness of vaccine side effects, unintentional unblinding may have occurred, and this can potentially inflate estimates of vaccine efficacy. Next slide. In conclusion, the potential benefits of this vaccine outweigh the identified risks. Therefore, I support the issuance of an EUA with the stipulation that the vaccine can protect against symptomatic disease. But at this time, it is not known if it prevents infection and transmission. Contraindication for those with a history of severe allergic reactions is plausible and should be included in the EUA as well. The continuing need for non-pharmaceutical interventions, particularly mask wearing, should also be noted. Finally, ongoing monitoring of this vaccine is imperative. Thank you.
Okay, the next speaker is Yvonne Fine.
Hi, my name is Evan Fine, and I'm a phase 1 participant in the Pfizer and BioNTech vaccine trial that was conducted by researchers at NYU. I have no financial or other conflicts of interest to disclose. I'm speaking today because after conversations with friends, family, and coworkers, I thought it was important to share my experience in this trial and to do my part to help ease people's fears about the vaccine. I think I got a real vaccine and not a placebo because of mild adverse effects such as fever, chills, and pain in the injection site after the second injection. After reporting the side effects, I was called repeatedly by the doctors and researchers at NYU to see if I was okay, and I was. Nothing felt rushed, and I never felt like a guinea pig. The question that everyone asks me is, well, what happened in the long term? Are there any long-term side effects? And it's been more than 5 months now since my first shot, and I can happily report that there are none. Since I've participated in the trial, I've helped out my older parents, I've gone to work in person, and I've exercised in small groups, and I haven't gotten COVID-19. I also want to emphasize that these activities do not represent changes of behavior that would alter the integrity of the clinical data. These are life activities that I have to do anyway. Not all of us are able to lock down and stay at home indefinitely. I'd still do my best to follow health rules and safety precautions. I understand the concern about whether or not people will trust the vaccine, but there will always be some holdouts. Most Americans will take the vaccine voluntarily as long as we're honest, don't talk down to them, and treat them like autonomous adults. But right now, demand for the vaccine exceeds the supply. The skepticism of some does not justify delays for others who desperately want to take it. The best way to get people to take the vaccine is to lead by example. I don't think that the problem was that this vaccine was rushed. The issue is that other important innovations are slowed down too much by delays in scheduling, lack of funding, and other bureaucratic rules that do little to enhance safety protocols. If Pfizer, BioNTech, Moderna, or anyone else has an RNA product that can immediately help people with cancer, dementia, or AIDS, the FDA should accept the data on a rolling basis and work with these companies to get to and complete phase 3 clinical trials within a year or two. 5 to 10 years for these trials is simply not good enough. Pfizer has just set the gold standard for future clinical trials. Let's live up to this. Now, this is an emergency, and the burden of proof is on those who don't want to authorize the vaccine. Absent compelling reason not to authorize it, it is simply immoral and unethical to deny the vaccine to healthcare workers or first responders who want it. An EUA must be granted, and it must be granted tonight. As of yesterday, the daily death toll for COVID-19 exceeds the death toll on September 11th, 2001. Delays on December 10th will mean more death on January 10th. In the words of Todd Beamer, let's roll. Thank you.
Thank you. The next speaker is Angela Rasmussen.
Thank you for allowing me a few moments to speak. I'm a virologist and affiliate at the Georgetown Center for Global Health Science and Security. I'm also, I have an advisory relationship with Siemens, but I have not been compensated to appear today. I'd like to offer some comments on how serology testing might better inform our knowledge of how these vaccines are working after they are rolled out to the public. So this expedited process has moved considerably faster than the typical vaccine development timeline, and as such, we are inherently limited on the breadth and scope of data that's available on duration, durability, and effectiveness of the immunity, as well as the variability of different vaccinees' respective immune responses. Due to exclusion criteria in the clinical trial itself, there's also limited data available on minority and underserved populations, as well as pediatric populations, pregnant women, and the elderly. And we don't really know a lot about how these broader populations will develop antibodies to the vaccines. With the limited supply of vaccines, at least initially, it's going to be really critical to determine who has already been infected with SARS coronavirus 2 in order to conserve the vaccine for equitable access for others in the same priority category. Because this rollout will be so highly visible and pivotal, we really need to address potential problems such as outbreaks in nursing homes, hospitals, schools, or other at-risk populations due to a failure to seroconvert. And serology testing could really help limit all of these issues. Next slide, please. Serology testing could help at different phases also before and after vaccination. So prior to vaccination, serology testing could assist in prioritizing individuals for vaccination, as I just mentioned, also establishing a serological baseline in the population, and help ensure that a scarce supply of vaccines initially reaches the most vulnerable people. Shortly after vaccination, it can confirm initial neutralizing antibody responses to the vaccine. It can also help ensure that antibody response clears the threshold for protective immunity. And over the longer term, 3, 6, and 9 months after vaccination, it can confirm the persistence and duration of immunity. It can also provide the means of bridging bridge trials to additional populations and subgroups. Finally, it can assess the protective efficacy of vaccination by distinguishing antibodies from vaccination versus antibodies from natural infection. So by looking for anti-nucleocapsid antibodies, you could actually determine if people who have been vaccinated have been infected after being vaccinated. And finally, annually after vaccination, we can assess better the persistence and duration of immunity. It can also inform the requirements for future vaccinations that are developed on an accelerated timeline such as this one. Thank you so much for allowing me time to speak.
Thank you. The next speaker is Jared Krupnick.
Your next presenter. Great, thank you. Before I begin, your next presenter, Dr. David Berger, alerted me that he can't get back into the conference. If you could resolve that for him, so thank you. So slide number 1, please. My name is Jared Krupnick. I'm the president of Uniting for Action and the founder of the Vaccine Considerations Project. I don't have any conflicts of interest to share. Slide number 2, please. As you can see, the Vaccine Considerations Project has been created to highlight health and safety throughout the COVID-19 vaccine evaluation process. I want to acknowledge and thank the incredible team behind all this work. Thank you, Dr. Eric Brown, Dr. David Berger, and our incredibly talented, committed, and rapidly growing team of graduate students. Slide number 3, please. I don't have to tell you about the challenges this committee and this agency have been facing. You're living those challenges. I'm bringing up the challenges to highlight that amid all the different reasons for vaccine hesitancy, what remains universal is that widespread concerns must be adequately addressed in order to ensure sufficient buy-in by public health experts, medical professionals, and the public. Slide number 4, please. You're seeking to establish trust and confidence in any vaccine you authorize and in the evaluation process by being rigorous, comprehensive, and transparent. There are limits to what can be addressed in a meeting. We're working hard to help you expand those limits. We're creating new ways to organize and share information. Here you can see how we've thoroughly analyzed the transcript of your previous meeting to extract specific concerns expressed by each individual committee member. We're providing the link to our website, vaccineconsiderations.com, where the full process transcript is available for anyone to see. Slide number 5, please. As you can see on this slide, our team has plotted specific concerns from specific individuals to develop a matrix of concerns. By plotting each concern as a row and placing each of the committee members in separate columns, you can use this matrix to quickly and easily see which members share concerns and which concerns have yet to be addressed or fully resolved. You can use this document as a living document where each member can continue to add and update their concerns offline, and the FDA staff and other committee members can address those concerns. By making this public, things won't fall through the cracks. This reassures the public and the professionals that are relying on you. The matrix of concerns is available to all at vaccineconsiderations.com. Slide number 6, please. We're using this spreadsheet as a model to demonstrate the concept, but a spreadsheet is limited. That's why we've spent over a year programming a custom platform called Information for Action that provides this type of functionality at scale. Our team is reaching out to stakeholders all around the country, experts, organizations, and associations to build and organize the central repository of concerns. We want to invite and encourage this committee and the FDA staff to utilize these technological tools to effectively address concerns, build trust, and reduce hesitancies. If you appreciate what we've shared, we'd like to connect with you. Please reach out to us at team@vaccineconsiderations.com. Thank you. My team and I look forward to working with you.
Great. The next speaker is Mr. David Berger, Dr. David Berger.
Hello. Thank you for allowing me to speak today. My name is David Berger. I am a board-certified pediatrician. I have no conflicts of interest. I am one of the few pediatricians in Florida who does not discharge families from my practice if they have vaccine hesitancy or do not wish to follow the recommended CDC schedule. My comments today are very mindful of the strong overall benefit our nation will get when most of us have immunity to SARS-CoV-2. Next slide, please. My presentation today is about vaccine hesitancy and steps that can be taken to increase my confidence in the COVID vaccine program. And this should be slide 3, by the way. It is very important that we see ongoing, complete transparency about the vaccine in terms of both safety and efficacy. We have seen reports that up to 50% of Americans and 30% of physicians have some level of hesitancy about COVID vaccines. We must allow for meaningful public scrutiny to build public confidence in the vaccine program. Next slide, slide 4. True informed consent has always been at the core of how I practice medicine. As doctors, we have a duty to inform our patients about the benefits and risks of any medical procedure. If a person feels she is not given sufficient information about a treatment, how can she provide informed consent? I also think it's important to respect people who have concerns about hesitancy about vaccines. I often find that hesitant families will proceed with vaccines if they don't feel that their concerns were blown off and minimized, but instead were respected and tended to. Slide 5. While there are many subpopulations that should be studied for increased chance of vaccine side effects, there is particular concern about those with pre-existing allergic, hyperinflammatory, and autoimmune conditions. The Pfizer data shows that 4 individuals who received the vaccine developed Bell's palsy, whereas there were no incidents in the placebo group. On the first day of England's vaccine program, their government had to tell people not to take the vaccine, the Pfizer vaccine, if they have a history of significant allergic reactions after 2 significant anaphylactoid reactions were seen in recipients. Slide 6. It will be difficult to quickly and fully track all 50 states to find the real frequency of adverse reactions. It is important we have a strong federal program to coordinate this. Please ensure a plan for this. Please provide data for us to know if people who have had COVID infection already have an increased or decreased risk of a vaccine reaction. Please provide comparative data between the different vaccine products to determine if any brand may have more or different reactions than other brands. Slide 7. I implore you to put in place a very long-term post-vaccine surveillance program. Many autoimmune and hyperinflammatory conditions often take a while to develop significant enough symptoms for a patient to seek medical help. Slide 8. It is also important to have a robust surveillance of COVID IgG antibodies so we will have a way of knowing if immunity is waning. We need to know if what is considered a protective antibody level for COVID-19, just like we have for other vaccine titers that can be commercially tested. Slide 9. If the FDA can increase our level of confidence, hesitancy will likely decline, and the COVID vaccine program will have a better chance of success. It will take many good people to defeat COVID-19, and I hope I can help you make a difference. Next slide. Thank you very much. I appreciate the time.
Okay, thank you, Dr. Berger. The next speaker is Sydney Wolfe, Dr. Sydney Wolfe, and he does not have PowerPoint.
Just like we have for other vaccine titers can be commercially tested. Slide 9. If the FDA can increase
Dr. Wolfe, you can't listen to it on the... Dr. Wolfe?
Yes.
Thank you very much.
Okay,
Can you hear me
you
with
gotta
the
turn your-
speakerphone on?
No, yeah, you have to turn your TV off or otherwise we're going to hear the delay. So please turn your TV off or whatever you're listening to.
I will turn it off right now. It's off.
All right.
Can you hear me
Dr.
on
Wolfe?
speakerphone? I'm doing
Yes, we can,
speakerphone
sir.
now.
Yes, we
Can you hear that?
Go ahead. Yes, we can. Go ahead, sir.
I'm Dr. Sidney Wolfe, founder and senior advisor of the Public Citizens Health Research Group. I have no conflicts. With the surging pandemic and interim efficacy and safety results made public 2 days ago, we now agree with the need for an EUA for the Pfizer-BioNTech COVID vaccine. There's an important unresolved conflict, though. If an EUA is granted for widespread use, should the 19,000 participants in the trial who received a placebo be notified of this and be offered a vaccine by Pfizer, clearly encouraging them to stay in the trial? Similarly, should blinded vaccine recipients be told of their status, thereby encouraging continuation in the trial? What would you wish if you were subjects in the trial? As status uninformed trial participants, you might otherwise leave the trial to get vaccinated with the Pfizer or any other available vaccine. The unblinding vaccine providing proposal has important advantages. First, once an EUA is granted, the ethical obligation, as Pfizer says, to both inform all placebo recipients of their status and offer them vaccine within the context of the trial is met. Second, by retaining more trial participants than if the trial remains blinded after an EUA, more recipients may be followed afterwards. The originally vaccinated group could be compared with the newly vaccinated group to continually compare rates of new COVID-19 infection with increasing duration of vaccination as well as adverse reactions. FDA has said that it doesn't consider availability, as you have all heard, of a COVID-19 vaccine under an EUA in and of itself as grounds for immediately stopping a blinded follow-up in an ongoing trial or offering a vaccine to all placebo recipients. During the October 22nd meeting, during Dr. Doran Fink's presentation, someone asked what about the problem of retaining placebo patients post-EUA, and Dr. Fink responded, with regards to mitigating the risk of dropout from ongoing clinical trials, we do share that concern. I don't have any specific remedies to offer at this time. We've asked the vaccine manufacturers to think carefully about how they would ensure clinical trial retention. Pfizer has stated in the briefing document, quote, it intends to continue the pivotal phase 3 study with participants in both the vaccine and placebo groups as originally allocated for as long as possible, emphasize that. Nevertheless, we have an ethical responsibility to inform all ongoing study participants of the availability and eligibility criteria of the vaccine made available under an EUA. They would accommodate those trial participants wishing to leave the trial by revealing which group they are in, but they can receive the vaccine only when practically eligible, depending on the government-specified priority group and the available supply. But Pfizer's real preference, and this is in the briefing documents, is that such placebo individuals are vaccinated within the study in order that both safety and efficacy data can continue to be collected. We believe this approach will minimize the number of current participants who withdraw from the study. If you were in the trial, would you prefer being unblinded only if you wish to leave the trial to seek possibly available but needed vaccine, or being automatically unblinded given Pfizer's vaccine if in the placebo group, or happy to find out
wrap it up
to stay in the trial? Thank you.
Thank you. The next speaker is Kim Woodsack.
Yes, Good afternoon. My name is Kim Woodsack, and I'm speaking on behalf of Woody Matters, a drug safety organization started after the death of my husband due to an undisclosed side effect of antidepressants. We represent the voice of families who live every day with the consequences of the current drug safety system. I am also on the board of directors for USA Patient Network, an independent patient voice advocating for safe, effective, and accessible medical treatments. Right now, the world is looking for hope, so they can get back to normal. Too many lives and livelihoods have been lost. Like many of us, we put blind faith and hope in the system that ultimately failed us. I have several concerns about rushing novel vaccines to market. The public needs assurances that the FDA's review was thorough and independent, given the process for reviewing new products usually takes 6 to 10 months. How could the FDA be as rigorous in weeks this time? Is there a process in place if there are dissenting scientific reviews within the agency? This needs to be available, and FDA scientists need to be protected for whatever their judgment is and not be political like it has been with many of the past controversies such as antidepressants and suicide has been handled within the agency. Transparency is everything. Assuming FDA approval, then all trial data must be released and made public. This also includes the process information like Pfizer's Data Monitoring Committee, who determined it was safe. Is it public? If not, it should be. We also need a transparent process for catching safety signals and communicating with the public. It needs to be real time like we saw in the UK yesterday with the allergic reactions, and not reported in weeks, months, years later like history has shown. Post-market monitoring will be more important than ever. We really don't know what short and long-term harms are given the short duration. Will FDA be staffed to handle the large task of closely monitoring that will be needed? Like MAUDE, the medical device reporting system, which includes patient narratives in the reporting, the FDA needs to do the same for the VAERS. Narratives can help tell a more complete story than just the data without giving away important patient details. Finally, I have huge concerns with possible unblinding of placebo participants and giving them the actual vaccine, as Pfizer CEO alluded to be willing to do. If this happens, we lose our control group. In closing, I'm directing this comment to the news media. You have a huge responsibility to dig deeper, ask critical questions, and not be just an extension of the manufacturer's PR department and accept press releases like the Wall Street Journal article did earlier this week and included a Pfizer supplied chart showing efficacy by subgroups where blacks had 100% efficacy. This should have sounded alarms and begged for additional questions, and then they would have dug deeper and realized there weren't a whole lot of blacks in the trial. Please seek out independent researchers, scientists, and others without political or financial agendas. Ultimately, the public is the real-world clinical trial. It is one big human experiment. The only ones that have 100% immunity in this will be the pharmaceutical companies. They get all the benefits of sales without any of the legal liability should something go wrong. I'd like to thank you for your careful consideration of my comments. I know firsthand the importance of your advisory committee work. Thank you.
Okay, the next speaker is Ms. Ligda D.
Hi, thank you. I'm from AIDS Action Baltimore and the AIDS Treatment Activist Coalition. I have no conflicts of interest. The answer to both agency questions on COVID-19 prevention and the risk-benefit ratio are clearly yes, in my opinion. Many of the issues submitted in my written comments have been addressed in the sponsor's submission. One important ethical issue involving maintaining participants on the placebo arm after EUA has been discussed. I think it is very reassuring that the sponsor is proposing that the placebo participants be permitted to decide whether to remain on the placebo or to receive the vaccine within the study at the appropriate time. The crossover design will hopefully ensure that the study will not crash. Crossovers are nobody's favorite, but an unblinded crossover is probably much more feasible here. Lay people have no understanding of the nuances discussed today. Acceptable designs that will confirm initial EUA data and protect current participants and suspicious future potential participants promoting public trust are essential. I do have a few important remaining concerns. The DART study for pregnant women should be much further along by now. Also, the sponsor is essentially claiming that safety and efficacy have been established across race and ethnicity. The analysis of 3,800 phase 2-3 participants includes only 0.5% Native Americans, 9.3 Black or African Americans, and 28 Latinx people. The Native American and Black or African American numbers are especially concerning in these key populations who are disproportionately affected by and who experience greater adverse COVID-19 related comorbidities and death. While my community appreciates the eventual inclusion of people with HIV, HBV, and HCV per Amendment 6, the mere 120 people with HIV enrolled is abysmal. The sponsor has submitted 0 separate data on people with HIV, HPV, and HCV, or people over 75. Thus, there is absolutely no data to guide vaccine use in these populations. This is especially concerning for people with HIV, as there is mounting evidence of disparate adverse co-infection outcomes. VRBPAC should recommend that the sponsor conduct post-EUA research to establish safety and efficacy in significant numbers of people over 75, in people of color, and people with HIV, HPV, and HCV where feasible, without excluding them from any indication. Finally, I hope we don't begin by lagging behind in the enrollment of people in color in COVID-19 research. I sincerely hope Pfizer will include consumers in its vaccine expert safety subcommittee and will convene community meetings to discuss trial designs as well as vaccine education and accrual and retention issues. This practice has served both the HIV community and sponsors very well over many years of successful and mutually beneficial drug development. Thank you and the FDA for such dedicated service and for the opportunity to comment.
Great, thank you. Next speaker is Peter Doshi.
Good afternoon. I'm Peter Doshi, president of the nonprofit Center for Science in the Public Interest and an associate commissioner at FDA from 2014 to 17. I have no conflicts of interest to disclose. I would like to thank the FDA for conducting its review in a transparent manner and for committing to the advisory committee review process, particularly under concerted political pressure. I just want to address 2 issues today. First, based on the data accrued to date, the Pfizer product demonstrates a striking degree of efficacy in preventing confirmed COVID-19, one that is shared across a variety of demographic, clinical, and other subgroups. Now, I agree with the FDA reviewers that there is no evidence of a major safety signal. However, the extent of more minor adverse events is notable. These include injection site reactions, fatigue, headache, all in over 50% of subjects, and chills in almost a third, all substantially elevated compared to rates in the placebo group. I do not believe that these events should stand between this product and authorization, but I do think the rates of these events are sufficiently elevated to merit open and even-handed discussion with patients. We are already facing significant levels of vaccine hesitancy, and if patients are not forewarned about these adverse events, the word will surely spread rapidly, potentially exacerbating the hesitancy problem. I also welcome FDA's identification of the disproportionate numbers of Bell's palsy cases and hypersensitivity-related adverse events observed in the treated group as matters that should continue to be monitored, including in the post-marketing phase. The second issue relates to trial design now that at least one safe and effective vaccine has been identified. The issues are complex, but we ought to be able to agree on this. No subject who has put their body on the line in a vaccine study should be at a disadvantage in terms of vaccine access as a result of their participation. Yet some observers appear to be advocating for extended periods of blinded follow-up even after authorization. This position is hard to justify ethically if it is inconsistent with public health recommendations at the time, particularly with rapidly rising case rates and the reported levels of effectiveness for the Pfizer and Moderna vaccines. So let me propose the following framework, which I think we can all agree on, at least most of it. 1, subjects should be informed if any vaccine candidate is authorized, not just the one in their trial. 2, like any study subjects, those in vaccine trials should be given the opportunity to leave the trial at any time if they so desire. 3, given the shortages of available product, subjects should be offered vaccination with an authorized vaccine as soon as it is offered to those in their clinical or demographic group in accordance with federal or state guidelines. 4, those for whom the product is not yet recommended can continue to be followed in blinded fashion. And 5, vaccination is recommended for an individual. A good option is to do so in a blinded crossover manner as described by Dr. Goodman to facilitate blinded follow-up for long-term safety and efficacy outcomes. I believe that this will facilitate the collection of essential data while honoring the contributions of the tens of thousands of people whose altruistic efforts have brought us to where we are today. Thank you.
Okay, thank you. The next speaker is Andrew Spiegel.
Yes, thank you. Thank you for allowing me the opportunity to provide comments on this most important day. My name is Andrew Spiegel, and today I present in my role as chair of the World Patients Alliance. I have no disclosures. The World Patients Alliance is a global umbrella organization of nearly 200 patient organizations from 86 countries representing hundreds of millions of patients across diseases worldwide. My very brief comments today fall into three basic areas: dedication, commitment to science and transparency, and innovation. First, we want to applaud the dedication of the scientists, both within the private companies and within the FDA. In the last year, scientists and researchers have fought tirelessly to create a vaccine for the novel coronavirus and put it through rigorous processes to ensure its safety and its efficacy. We are comforted in knowing that the FDA will require rigorous post-distribution data monitoring to continue to ensure any approved vaccines will be safe for the population. We know that in these unprecedented times, the people of America and the people of the world deserve to know that their governments are working to eliminate this threat. And we want to make sure that the FDA knows that we will continue to hope that it keeps the patients at the forefront of all of its policy and approval decisions on these vaccines as well as other medicines. The entire world is watching what the FDA is doing, and as everyone knows, they're relying upon the FDA's commitment to patients. Last, as the coronavirus vaccines continue to be approved and used in countries around the world, we must continue the process of innovation. Developing countries need access to innovative medicines now more than ever. In future breakthroughs, the vaccine for COVID could be manufactured as a pill that would be easily distributed and prescribed in those countries with less access to healthcare professionals around the globe. I certainly am in agreement with other speakers before me on the issue of ensuring diversity in clinical trials and ensuring trial members get access to the vaccine, and will not repeat further comments on that. Thanks again to all the stakeholders who have come together in an unprecedented way during these unprecedented times to do what would be unthinkable in the past. On behalf of patients around the globe, we thank you for your hard work and your commitment.
Okay, great. The next speaker is Misa Shafi.
Good afternoon. I'm Misa Shafi, and I'm here today on behalf of the National Consumers League. I have no relevant conflicts of interest. We extend our gratitude to the Vaccines and Related Biological Products Advisory Committee for the opportunity to present public comment today on behalf of consumers regarding the deployment of an emergency use authorization for the Pfizer-BioNTech COVID-19 vaccine. Firstly, we want to thank the Food and Drug Administration, Centers for Disease Control and Prevention, and other public health agencies for their demonstrated commitment to fostering public trust throughout the COVID-19 vaccine development and approval process. As consumer advocates, we have been encouraged by the honesty, transparency, and access afforded to the public during this critical time. To that point, there has never been a more critical time for consumers to have confidence in the FDA. The agency has undergone scrutiny from the scientific community for prematurely issuing EUAs for COVID-19 therapeutics. NCL is aware that developing a vaccine for COVID-19 is a time-sensitive priority and appreciates that the FDA recognizes that an EUA is not intended to replace long-term randomized clinical trials data associated with full FDA approval. We look forward to continuing guidance around the vaccine as the trial continues to collect safety and efficacy data 2 years post-release. We have great trust in the FDA's rigorous vaccine approval process and call on the agency to perform ongoing post-market surveillance to ensure the vaccine's continued safety and efficacy. Post-market surveillance performed in the UK yielded that the vaccine is unsafe for individuals with severe allergies. We call on the FDA to heed these warnings and to continue to sustain its robust interagency collaboration towards the evaluation, approval, and distribution of the COVID-19 vaccine. Consumers will rely on ongoing guidance from public health agencies regarding any potential adverse events from the vaccine. Additionally, ensuring innovative vaccine delivery methods, such as including oral or nasal options, could address geographic access issues as well as adequately consider diverse health needs, increasing overall uptake. We welcome efforts to ensure diversity in the clinical trials for the COVID-19 vaccine, and NCL requests that the FDA continue to prioritize vaccine clinical trial data that reflects diversity, as people of color will need to have confidence in the vaccine's efficacy in their communities. This will impact overall uptake of the vaccine. The development of a COVID-19 vaccine in such record time has been a miraculous feat, made possible through robust collaboration between private and public entities. NCL will continue to support the FDA and CDC in its effort to release a COVID-19 vaccine safely and expeditiously. Thank you to the committee for your consideration for our views on this important public health issue.
Okay, thank you. The next speaker is Julie Mahendro.
Good afternoon. I'm Julie Mahendro, unaware of any conflicts of interest. I will cover key points in the comments I submitted and leave you to read through those as you please. They were submitted under the banner of the Regulatory Watch Cat and easily identified by a cute black cat. I start with a question. Does the EUA violate Article 37 of the Declaration of Helsinki, which states that unproven intervention should not be used in medical practice without informed consent? This led to the questions of whether authorized products are proven or unproven, and whether their use is medical practice or research. Eventually, I concluded that EUA lies in an ethical gray area, falling along several continuums. I considered a few such continuums and ethical questions that they might raise. I also looked at ethical standards in 21 CFR, the Belmont Report, and the AMA Code of Medical Ethics. Finally, I considered Section 564 of the Food, Drug, and Cosmetic Act and the current status of both informed and consent for EUA products. Section 564 requires informing patients that the product has been authorized for emergency use. The problem is that patients don't know what that means. Currently, providers and patients are provided with product fact sheets, all apparently based on the same template. The language addressing the product has been authorized for emergency use comes directly from Section 564, which was not written to inform patients of anything. It is not in language understandable to patients nor to providers. Currently, there is much confusion among manufacturers, providers, and patients regarding the regulatory status of authorized products and what can reasonably be expected of their safety and effectiveness. It is the latter that patients and providers need to understand and where the fact sheets fall far short. If individuals with the appropriate expertise could evaluate concepts such as proven versus unproven, medical practice versus research, as they apply to EUA and as points along a continuum, this could provide a rational framework for addressing ethical concerns that have emerged with the EUA, as well as the appropriate regulatory oversight and the appropriate use of clinical data generated from the use of an EUA product. I don't think this type of evaluation is suited for warp speed. Therefore, in the interim, I ask the committee to consider that the risks presented by the EUA include not only ethical risks, but also loss of public trust, a serious risk for any public health program. If issues emerge with the use of a new medical product, patients and providers rarely go back to, what was I told? I hope FDA will give serious consideration to moving forward with any COVID-19 vaccine initially under expanded access rather than EUA to assure adequate ethical oversight, informed consent, and early access to the vaccine by the populations that most urgently need it. In addition, for the love of Harvey Wiley and Jonas Salk, please, please have an IRB take a look at the template for the patient fact sheets. Thank you.
Okay, great. The next speaker is Drew West.
Hi. Thank you. Thank you for this opportunity to comment. My name is Drew West, and I'm the president of the USA Patient Network. Our organization is composed of many patients and family members who have been affected by pharmaceutical and medical devices that promised hope, but left our members, our family members, harmed with lifelong side effects and sometimes death. We have no conflicts of interest to report, as we are a completely independent voice for patients and their families. Our concerns are based on lived experience. Every person receiving a COVID vaccine needs to know the answers to basic questions, such as, will this vaccine prevent the most serious symptoms, or the spread of the disease, what side effects or problems might occur immediately, midterm, and long-term, and how long will this vaccine protection last. It's our opinion that at this time there are too many unanswered questions regarding the safety and efficacy to release a vaccine. The release of these vaccines will make participants unwitting subjects in a seemingly uncontrolled clinical trial. That said, we recognize the tremendous pressure being placed on the FDA to act. Therefore, in the absence of reasonable assurances of safety and efficacy, we ask that the release of this vaccine be done cautiously and with thoughtfulness for the health and well-being of the recipients. We ask that you proceed slowly with release of these vaccines on a voluntary basis until full conclusion of blinded phase 3 clinical trials. We ask that you exclude populations that have not been studied or thoroughly studied, such as pregnant women, frail elderly persons, and severely immunocompromised persons, among others. We also ask that you continue or expand existing or new clinical trials to include groups of people who have not yet been included in any clinical trial to truly determine safety and efficacy. We ask that you monitor the vaccine recipients long-term over time by collecting and analyzing data to identify the real-world safety and adverse response results. This includes giving clear instructions and encouragement to vaccine recipients on what, where, and how to report adverse response events, not just reporting to their doctors. We ask that you give complete informed consent information that includes full disclosure of the ingredients in the vaccines, all possible adverse responses, as well as a statement in plain language that the current safety and efficacy information is incomplete and that the vaccines have not yet been studied for safety and effectiveness for everyone or studied for adverse responses or effectiveness over time. We thank you for hearing our concerns.
Okay, great. Thank you. The next speaker is Sarah Christopherson.
Hi, thank you. My name is Sarah Christopherson. I am the Policy Advocacy Director at the National Women's Health Network. We're a nonprofit advocacy organization that has been bringing the voices of women to the FDA for 45 years. We are supported by our members and do not accept financial support from drug or device makers, and I have no conflicts of interest to disclose. We applaud the FDA for their diligent work during this public health emergency on desperately needed vaccines, including, yes, over Thanksgiving. However, we do have serious concerns that moving forward with an emergency use authorization based on so little data for so many of the communities that have been hardest hit by this virus will do little to assuage legitimate concerns in those communities about taking the vaccine. As we heard this morning, CDC data indicate that Black and Indigenous people living in the U.S. are roughly 4 times more likely to be hospitalized from COVID-19 and roughly 3 times more likely to die from the virus than their white counterparts. And as we heard at the October 22nd meeting, members of those communities have also expressed a really strong interest in knowing that the vaccine will work in people like them. Without those assurances, Black Americans have expressed high rates of COVID vaccine hesitation, and for good reason. Black Americans don't have to look back to the last century or to the infamous Tuskegee study to see examples of the medical system undervaluing them. And many Black and Indigenous people have had multiple discriminatory and negative experiences with the medical system in their own lives. Pew Research found a clear link between confidence in the regulatory process that you all are pursuing and Americans' willingness to get the vaccine. Before authorization is granted, affected communities need to have confidence that the vaccine is safe and effective for people like them. The efficacy data submitted to this panel included fewer than 2,000 Black or African American vaccine recipients and just 131 American Indian or Alaska Native recipients, not counting the placebo control population. The safety data included just 206 Black seniors, 65 or older, and just 131 American Indian or Alaska Native people of any age. If this advisory committee votes to recommend authorization based on this data, FDA must ensure robust tracking and bold public transparency once the vaccine is taken. Otherwise, the near-term side effects of the vaccine, which have been mentioned earlier, such as fever, chills, pain, fatigue, all of which could be really quite alarming to the public not expecting it, could further fuel distrust among communities that have been given little reason to trust this process. Thank you.
Okay, thank you. The next speaker is Vicki Phillips.
My name is Dr. Vicki Phillips. I have no financial conflicts. I'm a public health scientist and nurse who has served as consumer representative on VRBPAC. I am the volunteer director of research and patient safety for the National Vaccine Information Center and the mother of a child injured by his 15-month well baby shots in 1998. The normal U.S. vaccine development, testing, and licensing process takes 10 to 20 years. When accelerated and when populations studied are not large enough or do not closely match those targeted to be vaccinated, the public assumes unknown and potentially increased risks. Under the emergency use authorization, COVID-19 vaccines can be approved but remain experimental and unlicensed, while manufacturers are shielded from liability if the vaccines cause harm. The EUA fact sheet prepared for the public must be transparent and fully disclose known and unknown risks of the Pfizer-BioNTech COVID-19 vaccine to guarantee fully informed medical decision-making and to ensure public confidence. Specifically, the EUA fact sheet should disclose whether the vaccine is at least 94% effective in preventing SARS-CoV-2 infection and transmission, or only prevents severe COVID-19 disease, hospitalization, and death, whether there is evidence for antibody-acquired and T-cell mediated immunity and how long it lasts, whether there is a risk of enhanced COVID-19 disease when vaccine recipients are exposed to the SARS-CoV-2 virus or have previously received flu shots or other vaccines, whether there is evidence for immediate and/or delayed serious vaccine reactions and poor health outcomes, especially in those who have already had COVID-19 disease, have severe allergies, experience severe reactions to previous vaccinations, or are chronically ill. The EUA fact sheet must clearly identify populations excluded from clinical trials. Over 40 criteria excluded certain people from some trials, including pregnant women and children, those with high blood pressure, obesity, diabetes, asthma, heart, lung, and kidney disease, neuro and immune problems, and those using certain medications or with a history of vaccine reactions or over age 85. Will the vaccine be safe and effective for them? The EUA fact sheet must clearly list all vaccine ingredients and disclose that an aborted fetal cell line was used to test the vaccine, as reported in a September 2020 paper published by 61 Pfizer and BioNTech scientists. By law, the EUA fact sheet must state that the consumer has the option to accept or refuse the vaccine. It is the position of the National Vaccine Information Center that using coercion and sanctions to persuade adults to take an experimental vaccine or give it to their children is unethical and unlawful. Thank you for your consideration.
Okay, thank you. The next speaker is Martha Nolan.
Good afternoon. Thank you for the opportunity to speak with you today. I am Martha Nolan, Senior Policy Advisor at Healthy Women. Healthy Women is the nation's leading nonprofit health organization representing more than 18 million women. We provide consumers and healthcare providers with accurate, evidence-based information about diseases and conditions, innovations in research and science, and changes in policy that affect women's access to treatment and care. I'd like to first thank you for the thoughtful and important work you are doing today and all that you've been doing throughout the pandemic. The discussion today is truly historic, and I commend all of you for your leadership and careful consideration of the data that is before you. Whether today or in the future, as COVID-19 vaccines are authorized by this body, I know that we are all cognizant of the challenges that lie ahead in terms of public trust. The science being presented today is truly remarkable, and the transparency of this meeting and the sharing of information from all of the companies as they work to develop COVID-19 vaccine candidates is the first step in bolstering public trust. As you're aware, recent surveys among the general public
but also somewhat surprising among healthcare workers, reveal concerns around the safety of COVID vaccines and many myths and misconceptions. When you look at communities of color, the level of skepticisms are even higher. All of us who work in public health must look to address this issue, and working together will be particularly critical. That is why Healthy Women is co-leading an effort with more than 60 other public health organizations representing patients, caregivers, and families, diverse communities, healthcare workers, older Americans, veterans, frontline workers, and scientists who work together to alleviate the concerns and hesitancy associated with the COVID vaccines. The COVID-19 Vaccine Education and Equities Project's missions is to educate and raise awareness of the importance of the COVID-19 vaccination for public health, the economy, and the broader society, as well as leading the conversation to ensure equitable access to authorized and approved vaccinations. The key to getting our lives back to normal here in the U.S. and across the globe hinge on the success of COVID-19 vaccinations. But we also know that vaccines are only as effective if they are trusted and taken by a majority of the population. We look forward to continuing our work to help build that trust and thank all of you again for the work you're doing to ensure that the coming vaccines are safe and effective. Thank you.
Okay, great. Thank you so much. The next speaker is Mitchell Warren.
Thank you so much. My name is Mitchell Warren, and I'm the Executive Director of AVAC, a nonprofit organization founded in 1995 to accelerate the ethical development and global delivery of HIV vaccines and other new prevention options. And in March, we joined with several organizations to establish the Global COVID Advocates Advisory Board. I have no conflicts to declare, and we accept no funding from pharmaceutical companies. AVAC enthusiastically welcomes the safety and efficacy data of the Pfizer-BioNTech mRNA vaccine candidate being presented today. This is terrific news to be sure, and a triumph of science and partnership. While the data to us do clearly warrant an EUA, they also require the maximization of additional data collection to address a number of remaining questions and issues that need to be addressed. One, the critical importance of distinguishing between an EUA and licensure under a BLA, and for you all to help ensure that continued data collection and a clearly articulated pathway and timeline for a BLA is provided. Two, there continues enormous need for the inclusion of diverse populations in COVID vaccine trials generally, and the data under review today provide limited information about the safety and efficacy data in diverse populations, including people living with HIV, other immunocompromised people, and those who are pregnant and breastfeeding. It is essential that specific requirements and timelines be articulated so that these key populations are not left behind. And I want to underscore my colleague Linda D's earlier comments on the critical importance of equity, diversity, and inclusion in research and review. With only 2 months of follow-up data as per EUA guidance, and it is essential that future BLAs include at least 6 months of follow-up. Therefore, continued blinded follow-up of the trial is warranted, while recognizing that some trial participants will want to exercise their right to leave the trial and if from the placebo group seek vaccination. If an EUA is granted, it will be urgent for the FDA and the companies to rapidly develop clear information, including an explicit reconsent process to outline the benefits, risks, and rights of maintaining in the blinded trial for both public health and personal benefit, and strongly encourage the committee to endorse the deferred blinded crossover design proposed by NIAID and presented earlier. In addition, we encourage the FDA to urgently issue guidance to other developers who will need to address this issue. Fourth, perhaps the biggest unknowns remain the durability of vaccine efficacy and whether the vaccine prevents asymptomatic disease and will limit transmission. A clear plan to collect and communicate information to inform answers from within the ongoing trial, as well as in the design of additional trials, is essential and should be clearly articulated when any EUA is announced, and should be strategically linked to other vaccine developers to jointly identify correlates, bridge data to other populations and platforms, and track use. As we've seen repeatedly in this pandemic and throughout HIV, clear evidence-based information is key. In conclusion, AVAC applauds this mRNA vaccine and both this VRBPAC and FDA for its commitment towards transparency, independence, and evidence-based scientific decision-making. This process is not just about authorization or approval. It is a beacon of independent review and transparency that will help to foster the trust necessary to rebuild confidence in vaccines, in science, and in our public institutions. Thank you for what you're doing today and throughout this process, and thank you for the opportunity to present, and looking forward to continued engagement with scientific and regulatory processes that move with speed of trust.
Okay, great. Thank you. And I think we are almost come to a closure. We tried the last person requested, and then we are unable to reach her out. So I think we can conclude because we already are past 10 minutes past the actual time for the next presentation. But before we go to sponsor presentation, the director of the Center for Biologics, Dr. Peter Marks, would like to make a few comments. Then we'll go to sponsor presentation. Thank you. Dr. Marks, you are on.
Thanks very much, Prabha. I just want to take a moment here to offer some thanks to various people. First of all, thanks to anyone from the public who's tuning in today. I think having this as a transparent process is very important as one of the steps we're taking to try to enhance vaccine confidence across the country. I also want to take a moment to thank all of the advisory committee members and the speakers today. Their contributions are immense, and we'll look forward to their discussion later on today. It's also very important for me to take a moment to thank a group of people at FDA that have put an incredible amount of effort into this advisory committee meeting while they're simultaneously getting ready for another advisory committee meeting and taking care of many other vaccines in various stages of development. The amount of work that has been put into this by those just setting up these meetings is tremendous, and I'd like to thank Prabha and all of the advisory committee staff, but also an incredible debt of gratitude to all of our reviewers in various offices, and especially in our Office of Vaccines Research and Review. The leadership and the staff in that office have worked tirelessly throughout the past weeks and months to get to this point, and that included working diligently over this past holiday when they could have been spending time with their families. So we're very, very grateful for all of the work and grateful to all of you. So with that, I want to let this proceeding move on since it's going to be a long day anyway, but thank you very much.
Thank you, Dr. Marks. Now, Dr. Mancho, back to you. Take on from this point on for the next presentation with the sponsor.
Right. Next, we have the sponsor presentations. The moderator is Catherine Jansen, Senior Vice President and Head Vaccine Research and Development of Pfizer, and William Gruber, Senior Vice President, Vaccine Clinical Research and Development of Pfizer. You're on.
Good afternoon, members of the committee, FDA, and ladies and gentlemen in the audience. It is a real pleasure to be here today. I'm Dr. Catherine Jansen, and I'm a Senior Vice President and Head of Vaccine Research and Development for Pfizer. I would like to thank the FDA for organizing the VRBPAC and the VRBPAC chair and members for their time. Pfizer and our partner BioNTech are pleased to be here today to discuss our candidate mRNA COVID-19 vaccine program in the context of requesting an emergency use authorization. Our presentation today will follow this agenda. After I give a brief introduction, Dr. William Gruber, Senior Vice President, Vaccine Clinical Research and Development, will review the development program for our vaccine, including non-clinical, clinical safety, and clinical efficacy data. After this, I will come back to review the benefit risk and provide conclusions for our presentation. We are also pleased to have a principal investigator with us to answer any study-related questions. Dr. Stephen Thomas, who is Professor of Medicine and Microbiology and Immunology, Chief Division of Infectious Diseases, and Director, Institute for Global Health and Translational Sciences from the State University of New York, Upstate Medical University. Our COVID-19 mRNA vaccine called BNT162b2 has been developed for the following indication: prevention of COVID-19 in individuals 16 years of age and older. The dose level is 30 micrograms with 2 doses given 21 days apart. The vaccine presentation is a 5-dose, multi-dose vial that is preservative-free and stored frozen between -80 and -60 degrees Celsius until use. Since the beginning of the year, we have been facing a devastating situation with COVID-19, the disease caused by the new coronavirus SARS-CoV-2. Tens of millions of people have been infected globally, and over 1.5 million people have already died. No one is safe from this disease, and certain groups, such as healthcare workers and first responders, the elderly, and people with underlying diseases, are at particularly high risk. It is now becoming very clear, particularly with the recent increasing rates of COVID-19 globally and in the United States, as discussed earlier by Dr. Hall, that we need a safe and efficacious vaccine to stem this devastating pandemic while also deploying other prevention strategies, such as infection control, including mask wearing and social distancing. SARS-CoV-2, the virus that causes COVID-19, is a new beta coronavirus that emerged late in 2019 in China. Shown in red are the coronavirus glycoprotein spikes, called S for short. The spike protein is an important vaccine target or antigen, as it mediates first the specific binding of the virus to the ACE2 host cell receptor, and then the fusion of the viral envelope with the host cell membrane. By these actions, the virus enters into human cells where it replicates, then spreads to other people, and often causing illness in the infected individual. Data available from other coronaviruses, such as SARS and MERS, had established that antibodies to S can block the binding of the virus to cells and prevent the infection of the human cell. So we chose the S protein as our vaccine antigen. Specifically, we chose the form of the S protein that was engineered to lock in the perfusion conformation, which is thought to be optimal for eliciting functional virus-neutralizing antibodies. We chose BioNTech's mRNA vaccine platform for a number of reasons, and given BioNTech's long experience with messenger RNA vaccines in the oncology space. mRNA vaccines are produced by cell-free production processes using chemically highly defined vaccine components, RNA and lipid. mRNA vaccines can induce broad immune responses, well suited to address a new pathogen in a situation where there's no or limited knowledge of what correlates with protection. The mRNA vaccine platform lacks unwanted viral or vaccine platform antigens that may lower a vaccine-induced immune response. As a consequence, mRNA vaccines can be boosted repeatedly, an important consideration when persistence of vaccine immunity is not yet known. Given that mRNA vaccines are essentially synthesized, they can be developed and scaled up quickly, a clear advantage over cell-based production processes when developing a vaccine in a pandemic setting. So how does an mRNA vaccine work? We have a deep scientific understanding of how such vaccines work. The mRNA codes, that means harbors the information for the vaccine antigen, which is a form of the S protein. The mRNA is stabilized to formulation with lipids to form what we call lipid nanoparticles, or LNPs. The LNP formulation is further optimized to allow efficient entry of the LNPs into human cells. This LNP is also optimized to be able to enter antigen-presenting cells that are efficient at stimulating a broad immune response, which we desired, given that we did not know and still do not know which response best correlates with protection. Inside the cell, the mRNA is released from the LNP. Just like any other messenger RNA in a cell, the mRNA from the vaccine can direct the synthesis of a protein by the cell's own production machinery. And since our vaccine codes for a form of the SARS-CoV-2 spike protein, that protein is also produced. Once produced, the whole S protein and its smaller fragments are presented to the human immune system that recognizes the protein as foreign, resulting in stimulation of T helper cells or CD4 T cells that activate other T cells and antibody-producing B cells. The antibodies can bind to the S protein on our SARS-CoV-2 and neutralize the virus, which means the virus is no longer capable of infecting a cell. Also produced are CD8 T cells that can eliminate virally infected cells. An important feature of mRNA vaccines is that they stimulate effective B cell and T cell memory responses. This ensures longer-term protection from viral infection and disease. An additional advantage of the mRNA platform is that the vaccine induces a Th1-biased CD4 positive T cell response that is linked to protection from viruses and thus minimizes a theoretical risk of vaccine-enhanced disease. The Th1 biased response is characterized by secretion of cytokines involved in fighting a viral infection, such as interferon gamma and IL-2. So in a nutshell, the immune responses induced by the mRNA vaccine are similar to what you may get in response to a viral infection. But, of course, the mRNA vaccine is non-infectious and cannot cause disease. When we started our COVID-19 vaccine partnership with BioNTech, we had the choice of a number of different mRNA vaccine candidates, different flavors of the mRNA, and different forms of the spike protein. Given the enormity of our mission, clinical data were important to us in deciding on the right candidate for a COVID-19 vaccine. So we evaluated not just one, but 4 different candidates in phase 1 to be able to make real-time scientific decisions to select the best candidate based on the following major selection criteria. With regard to safety, we were looking for the most favorable safety and tolerability profile in both younger and older adults. With regard to immunogenicity, we were looking for the broadest antiviral immune responses most likely associated with efficacy. And with regard to a rapid pandemic response, we were looking for the candidate that could be developed and produced most efficiently. Using these criteria, we selected BNT162b2, which contains a modified RNA that expresses a codon-optimized, full-length, pre-fusion stabilized spike protein of SARS-CoV-2. The top priorities for vaccine development in general and for a COVID-19 vaccine in particular are as follows. We must demonstrate that the vaccine is highly effective, meaning that it can help prevent COVID-19 in at least the majority of vaccinated people. We must demonstrate that the vaccine is safe with a robust safety data set generated in a very large pivotal efficacy study. And we also must demonstrate that we can consistently manufacture the vaccine to the highest standards. Throughout 2020, we have been asked often, how can this be done in a year or less, when the process normally takes many years? Well, vaccine development or anything else in a pandemic setting is not normal. It requires a completely different way of thinking and a strategy of parallel versus sequential R&D. Instead of waiting for preclinical and clinical data to emerge, substantial efforts and resources were poured into process development and manufacturing scale-up well before any clinical data were available. This has been an unprecedented investment early in development. In planning clinical development, we worked with the FDA and other regulators on a seamless trial which collapsed phases 1, 2, and 3 clinical development, also in an unprecedented way, with close to real-time communications and decision-making without ever stopping the trial. We are grateful to FDA and other regulatory agencies who have lent us their enormous support to work on these seamless development timelines. With substantial work on the mRNA platforms already done at BioNTech, we were able to quickly start clinical development and advance to phase 2-3 start at the end of July this year. And we had support from CDC to provide real data on regional SARS-CoV-2 and COVID-19 attack rates in the United States to optimize our selection of trial sites. Last, but certainly not least, with the dedication of investigators and site staff, as well as the motivation of tens of thousands of trial participants, we were able to enroll over 40,000 participants in a record 6 weeks, give them each 2 doses of vaccine, and ensure their safety while conducting the trial with strict compliance and uncompromising quality. And now, let me introduce for you the clear and compelling data package you will hear about today that we believe satisfy the FDA guidance for COVID-19 vaccine emergency use authorization. We have submitted all of the information required to meet the EUA guidance, and you will hear about them this morning. As for manufacturing data, FDA has reviewed the CMC data submitted to date for our vaccine and has determined that the CMC information is adequate to ensure the vaccine's quality and consistency for authorization of the product under an EUA. And now, Dr. Bill Gruber will discuss the non-clinical and clinical data sets with you, as well as our future plans.
Thank you, Catherine. It's my pleasure to share with you today the development program for our vaccine candidate, BNT162b2. I'm going to begin with a very brief summary of the non-clinical data that encouraged us to move forward into the clinic. This includes both toxicity studies as well as a study looking at a challenge model in rhesus macaques. These studies are described in the briefing document. 2 toxicity studies in rats, including the BNT162b2 vaccine construct, were completed with no safety concern. Development and reproductive toxicity studies are ongoing, with preliminary results available by mid-December. In a SARS coronavirus 2 rhesus challenge model, the BNT162b2 construct provided complete protection in the lungs as determined by nucleic acid amplification testing for SARS coronavirus 2 in bronchoalveolar lavage fluid. This information is now published. And importantly, there was no radiologic or histopathologic evidence of vaccine-elicited disease enhancement. Despite limitations of animal models, these findings anticipated results in our phase 3 clinical trial, in which there is no evidence of enhanced disease. These results were encouraging, satisfied FDA guidance criteria, and permitted progression of human clinical trials. I'm now going to share with you the clinical safety, immunogenicity, and efficacy data from our overall clinical development program. First, I will cover safety and immune response from the German and U.S. studies, and then cover aspects of the phase 2-3 trial finishing with safety and efficacy results. So let's begin with the two phase 1 studies. The German phase 1 dose-ranging study was conducted in individuals 18 to 55 years of age. 12 subjects received the active BNT162b2 vaccine for each dose level cohort. This study evaluated safety, binding and neutralizing antibody responses, as well as cell-mediated immune response to look at the potential for Th1 bias, CD4, and CD8 T-cell responses. The U.S. study is a seamless study where we had a phase 1 portion that moved into phase 2 and then phase 3. For the phase 1 dose-ranging portion, we included 18 to 55 and 65 to 85-year-old individuals, 12 of whom received vaccine and 3 received placebo per dose level cohort. We looked at safety and immunogenicity with both binding and neutralizing antibody responses and followed reactogenicity by electronic diary. These individuals will continue to be followed for a full 2 years after the second dose. The results from the phase 1 experience have now been published, and you have details in your briefing document. I'll just summarize for you briefly the reactogenicity from phase 1. Mild to moderate injection site pain was observed frequently and was consistent with local reactions observed with other commonly licensed and recommended adult vaccines. Fever and chills, along with other systemic manifestations, were observed. Reactogenicity was generally higher after dose 2 than dose 1, and reactogenicity events after each dose of the vaccine in older adults were milder and less frequent than those observed in younger adults. Now I'd like to summarize for you the antibody responses in phase 1 to 2 30 microgram doses of the chosen BNT162b2 vaccine, focusing on the neutralizing antibody titers from the U.S.-based trial. These results have been published in a peer-reviewed journal and are described in your briefing document. By day 28, or 7 days after the second 30 microgram dose, true 50% neutralizing antibody responses are observed. GMTs are shown at the top of each column. Antibody responses are well maintained out to day 52, approximately 1 month after dose 2, in both this younger 18 to 55 years of age group and the older 65 to 85 years of age group. GMTs are again shown at the top of each column. The GMTs in vaccinated participants range from 1.5 to 3.8-fold higher than the virus-neutralizing GMT of 94 observed in a panel of 38 convalescent sera labeled HCS. So this was very encouraging to us, that we were achieving a functional antibody response that could be associated with protection. It was also very important for us to examine cell-mediated immune response to be confident that we were getting a Th1 biased CD4 and strong CD8 T-cell response. These are data from the German trial. This first panel shows gamma interferon intracellular cytokine analysis data. We see a substantial increase in gamma interferon, very consistent with Th1, and at levels above responses following natural infection, labeled HC. If you look at the middle panel, you can see a relatively higher proportion of S-specific CD4 cells expressing gamma interferon, IL-2, or both, compared to a lower proportion expressing IL-4. So once again, this emphasizes a Th1 bias that could be associated with protection. And likewise, not only is it important to demonstrate the CD4 responses and the concomitant potential for inducing memory, it was important to demonstrate CD8 T-cell response to indicate the potential for virus killing of infected cells. And in the right-hand panel, you see a robust CD8 T-cell response that exceeds responses observed from natural infection, again labeled HC. So, on the basis of promising neutralizing antibody response, Th1 biased CD4 response, as well as the robust CD8 immune response, we were encouraged to move into the phase 2-3 portion of the study with the BNT162b2 vaccine construct. Outlined here for you at a very high level are the fundamental elements of the trial, where our goal is to enroll approximately 44,000 healthy subjects. Stable chronic disease is allowed because we find it important to make sure that those individuals with underlying diseases are included. They stand to have the greatest benefit from a vaccine because of their high morbidity associated with COVID-19. We also have included individuals with stable HIV, hepatitis B, and hepatitis C virus infections. At least 40% of participants are 56 years of age or older. This is important because we recognize that this population is also particularly vulnerable to severe disease. We also recognize the importance of conducting this study in people of color. So we have adopted an approach that assures a diverse racial and ethnicity profile, including Black and African American populations, Asians, and Hispanic Latinx populations. We've excluded immunocompromised individuals because it's yet to be determined if different dosing might be required in this population. And as you'll see later, we plan to evaluate those populations in future studies. So here are the demographics displayed for the full population data cut on over 43,000 subjects as of November the 14th, with good representation of gender, race, ethnicity, and age, with even splits between vaccine and placebo recipients. The age breakdown is highlighted to show the different age groups above and below 55 years old, and in the older 65 and above age groups. Note that over 9,000, or 20.9% of the 43,000 participants were over 65 years of age. So now let me share with you the safety data from our phase 2-3 portion of the clinical trial. I wanted to highlight something familiar to members of the VRBPAC committee. We have ongoing safety reviews by an independent data monitoring committee of unblinded safety data, and in fact, these are occurring weekly. This makes sense in the context of a rapidly enrolling trial with this new vaccine candidate. The DMC consists of 4 adult or pediatric infectious disease experts and 1 statistician, all with expertise in assessing vaccine safety, immune response, and efficacy. And the DMC, as recently as this past week, has identified no safety concerns during the duration of the clinical trial and has recommended that the study continue as planned at all of their safety reviews. This figure summarizes the safety database population submitted to the FDA for this review. Starting at the bottom, there are over 43,000 study participants with safety data collected in the trial as of the data cutoff on November the 14th. Moving up the figure, nearly 38,000 of these represent a subset with median safety follow-up time of 2 months post-dose 2, meeting FDA guidance. This means that there are over 19,000 participants for whom safety follow-up data is available for at least 2 months post-dose 2, shown in the second bar from the top. Of the total safety population, there are over 8,000 subjects, shown at the top, from whom 7 days of solicited local and systemic reactions were obtained by electronic diary. Shown here is a schematic of how safety of subjects was monitored. On the left-hand side at the top, vaccination doses were given 21 days apart. The first dose of vaccine was followed by very intense active surveillance for potential COVID-19 symptoms that would trigger a telehealth or in-person visit and nasal swab. This was done both as a safety measure as well as to evaluate efficacy. Individuals could either be swabbed at the investigative site or obtain a self-swab. We were very intent, of course, on otherwise tracking safety comprehensively And you can see at the bottom left of the slide that we used an electronic diary to address common reactions seen after vaccine administration, encompassing at least 6,000 subjects and at least 500 in each of the countries that are included in the trial. We also captured non-serious adverse events 1 month post-dose 2. We will actively collect serious adverse events for at least 6 months post-dose 2, and deaths and related SAEs out to the end of the trial at 2 years after the second dose. So now let me share with you electronic diary data related to local adverse events. This represents data that were captured over 7 days after dose 1 and dose 2 in 16 to 55 and 56 to 85-year-olds. I think you can quickly appreciate, looking at the two panels, which represent dose 1 and dose 2, that for redness, swelling, and pain at the injection site, the type of local reactions are very consistent to those seen with commonly licensed and recommended vaccines with very little redness or swelling. Pain was largely mild to moderate in severity. No grade 4 local reactions were observed. Again, a very satisfactory safety profile as far as local reactions are concerned. We also, of course, used the electronic diary to look at systemic events. The orientation of this slide is different in that we are looking first at events 7 days after dose 1, and those who received the vaccine on the top panel compared to those who received placebo on the bottom. Looking at both the 16 to 55-year-olds as well as the 56 to 85-year-olds, you can see that the reactions fall within a tolerable range compared to other adult vaccines. I would highlight fever and chills because these appear to be the most discriminating, as you can see, compared to placebo, but both were within an acceptable range. Now we are looking at systemic events 7 days after dose 2, and you can see a somewhat higher incidence of fever and chills, as well as other systemic manifestations compared to placebo. The only grade 3 or severe solicited adverse events greater than or equal to 2% in frequency after the first or second dose were fatigue at 3.8% and headache at 2% following dose 2. One vaccine recipient reported a fever of 41.2 degrees centigrade only on day 2 after dose 2 and reported no fevers for all other reporting days. One vaccine recipient reported fever of 40.7 degrees on day 4 after dose 1 with no fever at the end of the 7-day reporting period. Otherwise, no grade 4 systemic reactions were observed. There was a difference between younger individuals and older individuals. Younger individuals tended to have more reactions. But in all age groups, the vaccine was well tolerated and the reactions were within an acceptable range. If we consider how these events peak and decline over the 7-day post-dose 2 period in the BNT162b2 group, it can be seen that the duration is short-lived. Participants are vaccinated on day 1. Fever, the leftmost dark blue bar, typically appears on the day after vaccination and lasts only a single day. Otherwise, systemic events, shown as the other colored bars, peak at day 2, then rapidly decline over the next 2 days in both age groups. We captured spontaneously reported adverse events by system organ class in the nearly 38,000 subject subset for which median safety follow-up was 2 months after dose 2. This slide shows adverse events by system organ class occurring in 1% or more of the study population. More details are included in your briefing document. The most common adverse events observed were general disorders and administration site conditions. As shown in the footnotes, the top 4 classes of unsolicited reactions in this nearly 38,000 participant data set mirror the common reaction captured by electronic diary in the 8,000 participant subset previously described. For example, these include reports of injection pain, fever, myalgia. For nervous disorders, the highest proportion is headache. For GI disorders, not diarrhea and vomiting. When we exclude those terms reflecting local reactions and systemic events typically occurring within 7 days of vaccination, we see a more even split of adverse events between the active vaccine and placebo. Apart from general disorders and administration site conditions where the predominant remaining event is unspecified pain in the vaccine group, 2.4% versus 0.2%. In general, adverse events by system organ class are infrequent and within range of such reactions reported after other licensed vaccines. Rounding out this part of the safety review, serious adverse events shown here by system organ class are consistent with what we typically see in populations that not only include 40% of individuals being older than 55, but over 50% of the population being obese and or having at least one underlying comorbidity. SAEs have been balanced between vaccine and placebo recipients. These include observed serious adverse events of special interest designated by the CDC, which are few in number and comparable between vaccine and placebo recipients. A total of 6 deaths have occurred in this population, with 4 of these in the placebo group. None of these have been considered related by the investigator. Further description of these deaths and the full safety data available as of November the 14th are included in your briefing document. So in summary, the tolerability and safety profile of the BNT162b2 vaccine at 30 micrograms administered as a 2-dose regimen 21 days apart is favorable, and no clinically significant safety findings other than mostly mild or moderate reactogenicity have been identified. I'd now like to turn to our efficacy evaluation. So let me now summarize how we went about determining efficacy for the first primary efficacy endpoint. Again, the vaccine doses were administered 21 days apart. And to qualify for the first primary efficacy endpoint evaluation, individuals needed to have no evidence of prior or current infection before each dose. And that was determined either by obtaining a swab at the time of each dose to identify evidence of SARS coronavirus 2 by nucleic acid amplification testing, or obtaining a blood specimen for N-antigen antibodies at the time of the first dose to indicate evidence of prior infection that may have preceded vaccination by months. So this allowed us to be confident that the individuals for the purpose of this primary endpoint had no evidence of prior or current infection at the time of each dose. And again, this is important because this is the group that we would all anticipate is most vulnerable to SARS coronavirus 2 disease or COVID-19. And then, of course, as I already mentioned, we have active surveillance for potential COVID-19 symptoms that trigger a telehealth or in-person visit and a nasal swab. And we will continue to do this for up to 2 years after the second dose. So now let me summarize for you what constitutes a case definition. To qualify as a case for the first primary endpoint case definition, individuals had to be baseline negative by serology and PCR for prior or current infection. And then we characterized the illness as needing to include one or more of these symptoms. And these should be familiar to you because they largely coincide with symptoms that are captured by the CDC case definition, but with a bit more potential specificity. Comparable efficacy observed encompassing all the CDC criteria are shown in the briefing document. And then on the right-hand side, once an individual qualifies for those first 2 categories, they need to have a positive validated PCR, either in our central laboratory, or results will be accepted from a local laboratory if it's approved as a type of testing that we agree is valid. All testing was performed blinded to treatment assignment. And so it's this combination that determines the case definition for the efficacy results that I'll now be sharing with you. We performed an interim analysis at 94 cases in individuals without prior infection and observed efficacy of 95.7%. We have now also performed the final vaccine efficacy evaluation against COVID-19 occurrence from 7 days after dose 2 in 170 cases without prior evidence of prior infection. Observed efficacy is high at 95% with high confidence based on the parameters shown in the 2 right-hand columns. There's 95% probability that efficacy falls in the interval shown, meaning over 97.5% likelihood that efficacy is greater than 90%. Likewise, the probability that vaccine efficacy is at least greater than 30% greatly exceeds FDA COVID-19 vaccine guidance. This efficacy trial is not powered to evaluate efficacy based on age, stratum, gender, racial, or ethnic group. Nonetheless, we think it would be useful for the VRBPAC committee to see vaccine efficacy broken down by these parameters. As you can see, observed efficacy was high, regardless of age, and consistent with overall results. 15 cases were seen in adults 65 to 74 years of age, and only one was in the vaccine group. 5 cases were observed in participants greater than or equal to 75 years of age, and all were in the placebo group. Likewise, as shown on this slide, efficacy was high in both males and females, and efficacy was high across racial and ethnic groups. Comparable high observed efficacy was observed across white, black, African American, other racial groups, and likewise also across Hispanic and non-Hispanic ethnicity, with lower bounds of the confidence intervals above 80% across these ethnic groups. There were also comparable values of observed efficacy seen across geographies. This efficacy trial was also not powered to evaluate efficacy based on risk groups. Nonetheless, we think it would be useful for the VRBPAC committee to see vaccine efficacy broken down by these parameters. The risk groups included individuals with body mass index greater than or equal to 30 kilograms per meter squared, and or those with Charlson comorbidity index, which included malignancies, chronic pulmonary disease, chronic cardiovascular disease, diabetes, renal disease, and many others. As you can see, observed efficacy was high regardless of whether the participants were at risk or not, consistent with overall results. Likewise, as shown on this slide, efficacy was high across age groups with and without risk, as well as those with or without obesity. If we break out comorbidity, we see that regardless of category, past history of malignancy, cardiovascular disease, pulmonary disease, diabetes, or additional category of hypertension, point estimates of observed efficacy remain high, and for some, the nominal lower bound of the confidence intervals are well above 0. Hence, we can be confident that the vaccine is likely to work well in older or debilitated individuals. Likewise, we have now evaluated efficacy against COVID-19 from 7 days after dose 2 in those with and without prior infection, the second of our 2 primary endpoints. Efficacy remains high at 94.6% with similarly high confidence based on the parameters shown in the right-hand columns. It was also important for us to define severe cases both for evaluation of safety and for determinations of efficacy. And for this, we use the definition of severe COVID-19 based on FDA guidance, and that's summarized here for you in bullet form. ICU admission, clinical signs of severe disease, organ or respiratory failure, and death are key features. Using the FDA definition of severe disease, let's first look at the top table. Although not statistically significant due to a small number of cases, protection against the few cases of severe disease occurring at least 7 days after dose 2 is consistent with the overall efficacy results, with 1 case in the vaccine group and 3 cases in the placebo group in those without prior infection, shown here. However, if one examines the all-available population for first severe COVID-19 cases after dose 1, the bottom table, only 1 case is seen in the vaccine group and 9 cases are observed in the placebo group for an observed vaccine efficacy of 88.9%. The vaccine recipient only met a single FDA criterion for severe disease of O2 saturation below 93% and was not hospitalized. In contrast, out of the 9 placebo recipients with severe disease, 6 met 2 or more criteria, 6 were hospitalized, 3 were admitted to the ICU, and 1 was intubated or mechanically ventilated. This is consistent with overall vaccine efficacy seen 7 or more days after the second dose and indicates the BNT162b2 vaccine is likely to protect well against serious disease. The FDA definition does not include hospitalization as a specific criterion for severe disease. However, the CDC definition of severe disease, shown in the light blue box, includes hospitalization, more severe outcomes of hospitalization, and death. We thought it would be useful to perform a post hoc analysis of severe disease using the CDC definition to further assess the impact of vaccine on this outcome. Using this parameter, efficacy against severe disease greater than or equal to 7 days after dose 2 was observed, with 0 cases in the vaccine group and 5 cases in the placebo group, with confidence interval as shown. Once again, protection was also observed for first severe COVID-19 occurrence after dose 1 of 92.9% with a 1 to 14 case split and lower bound of the 95% confidence interval well above 0. One vaccine recipient was hospitalized 12 days after receiving the first dose of the vaccine, but without additional CDC-defined morbidity. In contrast, of the 14 placebo recipients hospitalized, 3 were admitted to the ICU and 1 was intubated and mechanically ventilated. This analysis provides further evidence for vaccine protection against hospitalization and attendant morbidities. This curve shows the cumulative incidence of all available COVID-19 cases beginning after dose 1. Placebo cases are in red, vaccine cases in blue. Darker dots represent severe cases using the FDA guidance definition, 9 in the placebo group and 1 in the vaccine group, with 2 instances where cases overlap graphically at day 8 and day 67 in the placebo group. One can see that by at least 14 days, the curves begin to spread, indicating some efficacy after the first dose. Placebo cases continue to increase out to 105 days at the time of this data cut, while the vaccine case curve remains relatively flat. One can see in this expanded view from dose 1 to day 21 that the curves begin to spread by as soon as day 12 and at least by day 14 after the first vaccine dose. This graphic provides
hold on a second, Pfizer. We are not hearing you at the moment. So let's just take a moment here. Let's just check on Pfizer's connection. Just out of curiosity, can I get a sound check from somebody else, please?
Can you hear me?
That's test. Okay, thank you. That's what I wanted.
This is Archie.
All right, so hold on a second.
It's them, it's not us.
Yep, I figured as much. Pfizer, we are going to reach out to our sponsor here and tell them that they have to connect their audio. They just looks like their backup dropped. So Pfizer, please go to your backup audio. Here it comes. Are you there, Pfizer?
We're here.
Yep.
All right, there you go. So, sir, just go back about one slide, just to the beginning of this slide, please. We don't want to lose any content, okay?
All right, thank you.
Not a problem.
So apologies, everyone. Are we ready
all right,
to
so
roll?
we'll hand it back to, we're going to hand it back to Pfizer whenever you're ready.
I'm ready. Okay?
All right, take it away. Yep, take it away.
Okay, great. Thank you. Sorry for the technical difficulties. So our efficacy conclusions are as follows. Both primary efficacy objectives met the success criteria. In individuals without prior SARS coronavirus 2 infection, observed vaccine efficacy against COVID-19 occurring at least 7 days after dose 2 was 95%, with high probability, 97.5%, that the true vaccine efficacy is at least 90%. And again, this meets the pre-specified FDA criteria for emergency use authorization. Observed vaccine efficacy was greater than 93% for the first primary endpoint across age, race, ethnicity, and at-risk subgroups. Per the FDA definition, 9 severe COVID-19 cases were observed in the placebo group and 1 in the vaccine group as of the interim analysis cutoff dates, and 14 hospitalizations and associated morbidities were seen in placebo recipients versus 1 vaccine recipient hospitalization in a post hoc analysis, providing further evidence to support efficacy against severe disease consistent with that seen against all COVID-19. From the cumulative incidence curve, there is early onset of protection with divergence of the placebo group from the BNT162b2 group as soon as 12 days and by at least 14 days, with steady accumulation of cases in the placebo group while the vaccine group remains virtually flat. Overall, the efficacy results show that the vaccine at 30 micrograms provides protection against COVID-19 in participants who had or did not have prior SARS coronavirus 2 infection and COVID-19. Our emergency use authorization application is based on our satisfactory safety profile and efficacy against COVID-19. The emergency use authorization will be seeking an indication for individuals who are 16 years of age and older. This safety data includes reactogenicity in over 8,000 individuals, adverse events and serious adverse events in over 37,000 individuals with a median of 2 months of follow-up post-dose 2, and adverse events and serious adverse events in over 43,000 individuals 16 years of age and older with varying degrees of follow-up. The BLA, which we plan to submit in 2021, will encompass reactogenicity in over 8,000 individuals and AEs and SAEs in at least 44,000 total participants. Of these, safety data will be provided from at least 6,000 participants with 6 months or more of safety follow-up post-dose 2. Additional data will include safety in a 12 to 15-year-old population, which is currently undergoing study as part of our current phase 2/3 trial. The efficacy that we have presented to you today is based on more than 164 SARS coronavirus 2 cases and immune response in adults 18 years of age and older. This also serves as the foundation of data for the BLA, which we plan to file by April of 2021. Additional planned analyses include efficacy against asymptomatic infection, evaluation for persistence of protection, and a 12 to 15-year-old immunobridging study. As you will note in the Pfizer briefing document, we plan to administer vaccine to placebo recipients. We have an ethical obligation to inform study participants of COVID-19 vaccine availability under emergency use authorization. We are currently in discussions with the FDA about the best way to vaccinate placebo recipients to further inform safety and efficacy of the vaccine while meeting our ethical obligations to participants. Eligible participants in the placebo group will have the option to receive the vaccine. Since it will take time to provide vaccine to over 22,000 placebo recipients, we plan to first offer vaccine to those individuals satisfying FDA emergency use authorization and current CDC recommendations. Vaccination of other participants will expand over time. Participants will be given the option to remain blinded if they choose to do so through study completion. The study will continue for the planned 24 months regardless. Pfizer plans to meet FDA EU FDA EUA guidance for risk and benefit during use of the vaccine under the emergency use authorization. First, let's talk about pharmacovigilance. Since this vaccine is likely to be administered to millions of people in a short time, we've expanded our capacity to process AE reports with an online adverse event reporting portal. Our signal detection activities will occur on a more frequent cycle. We have plans for future clinical studies to expand to the more vulnerable populations. Second, proactive risk minimization. Comprehensive labeling and educational materials for vaccine providers will emphasize key messages about appropriate handling, storage, and preparation of the vaccine. And for vaccinees, emphasize the importance of following up for their second dose to maximize their protection. Product and cold chain will be monitored in real time. Third, pharmacoepidemiology studies. Several safety studies are planned to continue safety data collection. These will access healthcare information from millions of lives to monitor safety events, including adverse events of special interest. We know our vaccine works in the clinical study setting and plan to investigate its effectiveness in real-world use. And finally, collaborating with vaccine safety stakeholders. At a previous VRBPAC and this morning, we've heard about the FDA and CDC's planned programs for pharmacovigilance, including VAERS, CISA Network, Be Safe, and BSD, and we've shown you some of our plans, which are intended to be complementary to CDC and FDA planned pharmacovigilance activities. After approval, Pfizer will conduct test-negative design studies to demonstrate real-world effectiveness against severe disease with important endpoints like hospitalizations and emergency department visits in specific populations, and to understand vaccine efficacy when vaccine is used in real-world conditions and in broader populations. These studies will complement the CDC planned effectiveness studies. We also intend to conduct a number of studies recognizing that there are other populations that stand to benefit, and there are other things that we need to learn about the vaccine. These include boostability, studies in pediatrics and pregnancy, and use in immunocompromised populations. We also plan to explore a refrigerator-stable next-generation formulation and concomitant use with influenza vaccine. We look forward to expanding the safety, immunogenicity, and efficacy profile demonstrated today. So now that ends my summary of the clinical development program, and I'm pleased to turn our presentation back over to Dr. Catherine Jansen.
Mr. Gruber, we believe that our data have satisfied the EUA requirements for a COVID-19 vaccine, as you see here on the green check marks. We are seeking emergency use authorization for our vaccine for prevention of COVID-19 caused by SARS-CoV-2 in individuals 16 years and older, with or without evidence of prior infection with SARS-CoV-2. We have demonstrated a positive benefit-risk profile for our BNT162b2 vaccine, given the high overall vaccine efficacy of 95%, with high efficacy observed in both younger and older adults, as well as diverse demographics with and without underlying comorbidities. And we observed efficacy against severe COVID-19. In addition, we have demonstrated a favorable safety and tolerability profile in more than 40,000 individuals. So why are we applying for an emergency use authorization for BNT162b2 now? With the high efficacy and good safety profile shown for our vaccine and the pandemic essentially out of control, vaccine introduction is an urgent need. Dramatic increases in cases have occurred all over the country, and it is estimated that about 55,000 deaths will likely occur every month over the next few months. Modeling from the CDC shows that a vaccine with high efficacy can save many lives. However, a pandemic vaccine must be introduced before the peak of cases impact, which is best achieved under EUA. We at Pfizer and BioNTech wish to thank all of our sites, investigators, and their dedicated staff. And we want to thank our clinical trial participants, without whom we would not be here today. We are also grateful for the guidance provided by the CDC, the FDA, and other regulatory bodies and members of Operation Warp Speed. Finally, we want to thank our colleagues at BioNTech, Pfizer, and other companies for their tireless work and dedication to develop our COVID-19 vaccine candidate. Thank you for your attention, and we would be happy to take any questions.
Thank you very much to both of you. I wanted first to ask Dr. Gruber about how he is going to be measuring asymptomatic infections. In addition, I wanted to let everybody know that Bill Gruber is not related to Marion Gruber. So, Bill?
That's correct. Not to my knowledge.
All right. If I understood you, heard you correctly, because there was a little bit of a delay, you were asking about our plans, how to monitor asymptomatic infection.
Correct.
So indeed, our protocol, our trial, was actually designed not just to look for symptomatic COVID-19, but also to monitor and explore whether our vaccine is efficacious against asymptomatic infection. And we are monitoring individuals and screening them with a serological test that measures exposure to SARS-CoV-2. It's actually an N protein test.
Right, the N protein.
We hope that we will have those analyses completed very soon in the new year.
And you are also looking then at correlates of protections.
That is correct. In our large study with collecting serological samples throughout the study up to study conclusion, which is 24 months duration, we have the opportunity to look for a correlate of protection, provided that we see enough breakthrough cases over time. That's right. So, of course, with a vaccine that is highly efficacious, and if it continues to be highly efficacious over time, it may be a little bit more difficult to define a correlate of protection, but nevertheless, we are trying to do that.
Okay, we have many, many questions, and we're going to have to limit them and hope you and expect you to stay on for our open discussion in about an hour or so, because I think we can get to some of the other issues at that point. So, Dr. Levy.
Hi, thank you. I'm Dr. Ofer Levy with the Precision Vaccines Program at Boston Children's Hospital. I'd like to thank Pfizer for its impressive effort and excellent presentation. I have the following questions. For Dr. Jansen, one regarding mechanism of action. RNAs in principle can be recognized via the innate immune system, specifically potential RNA-induced activation of toll-like receptors 7 and 8 could induce interferons, which may explain the transient lymphocytopenia that was observed in the study and may provide an adjuvant effect, contributing to robust immune responses and the protection observed. Recognizing that the vaccine contains modified RNA that may have blunted innate immune activating potential, what is known regarding the ability of the lead mRNA to activate toll-like receptors such as TLR7 and 8? Has the mRNA vaccine-induced TLR activation been assessed in vitro in cell culture? And my questions for Dr. Bill Gruber Regarding the safety, the briefing document alludes to numerical imbalances of AEs potentially representing allergic reaction. What is Pfizer's knowledge regarding any potential vaccine AE relating to allergic reactions from either animal models or human data? And finally, regarding immunogenicity, Dr. Gruber described the adaptive response. What about the innate? Were plasma cytokines, chemokines measured, and did these correlate with any of the study endpoints? Thank you.
Yeah, so I would like to actually have Dr. Sahin describe in detail what we know about the mechanisms of actions. Let me just start out while Dr. Sahin is getting ready to say that one of the reasons that we shows the modified RNA platform for our vaccine is the reason that indeed it tempers, if you want, tunes down, if you want, the innate immune system. Because as I mentioned earlier, we were looking for a candidate that would allow us to have a very, or would give us a vaccine with a very favorable safety profile. If I could ask Dr. Sahin, please, to comment on the more detailed questions about signaling pathways for the mRNA vaccine candidates.
Yeah. Can you hear me? It's Ugo Sahin.
Yes.
Yeah.
Yes.
Thanks, Catherine. This is indeed an excellent question. So the nucleoside modified mRNA does not directly activate TLR7 and TLR8 in the human setting. And the adjuvant activity is driven by a combination of the recognition of messenger RNA components plus the lipid nanoparticle formulation, which disrupts the integrity of the plasma membrane, which integrates innate immune stimulation signals that result in activation of the type 1 interferon pathway, and this results in cytokine secretion, resulting in the lymphocyte recirculation from the blood into the peripheral tissue.
Thank you. Bill, you want to touch on the other part of the question?
Sure, Catherine, and we can obviously, if there are additional questions, follow up in terms of pharmacovigilance. But I think the first question was about adverse events within the study. Amongst the 44,000 subjects, we saw no serious allergic reactions to the vaccine. There were no substantive differences in standard MEDRA queries in numbers between the BNT construct and the placebo groups analyzed on the 38,000 subject database. So within the clinical trial, we've actually not seen evidence to suggest a signal related to allergic reaction to the vaccine. Obviously, we're conscious of the report that's occurred with use in the UK, and perhaps Patrick Cabell can speak to that. As far as and maybe while he's preparing, let me just deal with the second part of your question, which was the nature of cytokine measurement and the like. The measurements that were obtained were the ones that I shared with you, and I think actually as of today, it's my understanding that this is now being published from the phase 1 trial, from the German trial, some of which I shared with you in talking about the CD4 responses. But we have not, as part of either our phase 1 trial in the United States or the phase 2/3 extension, looked specifically at cytokines as part of that trial.
Thank you.
Thank you.
Thank you. Dr. Moore.
It's very exciting data. Thank you very much. Perhaps the most interesting data I've heard in this epidemic. I have two questions for you. One is related to Oprah's question, which is the modified RNAs in this vaccine. Do we know anything about whether the modified RNAs undergo salvage, could be reincorporated into, for instance, DNA, or if cellular reverse transcriptases could possibly amplify them or turn them into DNA? That's one question. Second one is, among the 8 people that were vaccine failures, do you have were you able to sequence the virus that came from those people, and was there evidence of antibody escape from your antigen?
Yeah, so I will start with answering your last question, and then I'd like to get Dr. Phil Domitzer to be ready to address your first question. So as of the 8 individuals that had a vaccine failure in our study, we have not yet analyzed, for example, immune responses or anything else for those individuals. But we plan to do this, but we don't have the data today. And if I could ask Dr. Domitzer to answer the question that you had about the RNA.
Yes, I guess there were 2 questions. One is, are the modified nucleosides recycled into other RNAs? And second, about the possibility of reverse transcriptase transforming the RNA into DNA. We do not have data on recycling of the modified nucleoside. Although it's theoretically possible for a reverse transcriptase to act on RNA, we think that the probability is actually quite low that this would occur. These RNAs have no signals that would make them preferential substrates for reverse transcriptase. They're present transiently in a relatively low amount, and we actually have very recent data indicating that actually it's a relatively modest amount of RNA that gets into the cell, and we actually don't see it getting into the nucleus where this would occur. So there doesn't seem to be any greater likelihood that a vaccine RNA would end up reverse transcribed than a cellular RNA.
Thank you.
Okay, thank you. Dr. Offit.
Yes, thank you. So this question is probably for Bill. First of all, thank you for an excellent presentation. The one thing that's been raised is that there roughly were 162 participants who got sick that were in the placebo group, which would mean a disease rate of about 0.74%. It seems like that might be low. But so it would be interesting to know whether or not that was lower than the disease rate in the area where these participants were being recruited, because the concern is that if that is true, if it is lower than typical, that we may have an example of volunteer bias, where people who volunteer for this trial are more likely to physically distance, more likely to wear a mask, and therefore more likely to be exposed to a lower inoculum. So could you comment on that? Was that 0.74% disease rate lower than you were seeing in that area, or was that typical for the areas where you were recruiting?
Bill?
Yeah, I think, of course, it's a good question, Dr. Offit. The rate of disease that we were seeing, I think, was relatively typical of not only the area, but thinking ahead to the nature of this population. On the one hand, this is a population that we recruited because they were at increased risk, but they also were voting with their feet to be part of a vaccine trial. And hence we assume that, of course, they were, and we certainly encourage them to take the appropriate sorts of social distancing measures and masking. So I expect it was probably some combination of the two. The other thing to keep in mind is that this trial, although it's been a very, short duration in terms of being able to get to our efficacy endpoint, did span a period of time, particularly in the United States, where rates dropped. And then, of course, we now know the horrific situation we're in now with over 3,000 deaths a day. But it's my sense that part of this is the nature of the span of the trial, both during a time when the rates were high and low, as well as the precautions that individuals in the trial, even though they were high risk, took to further reduce that risk.
Thank you.
Thank you.
Okay, Dr. Pergam.
Thanks. First of all, I just want to thank Pfizer again for, first of all, presenting this data. And I want to really thank them for providing this data to the public as well, because I think the way that they presented this data to the community was really important for many different reasons. I've been curious about enrollment related to risk categories. The large proportion of people in this trial are white, and there's about, I think I characterized 46% had the comorbidity, and a certain number were of older age, et cetera. My curiosity is about timing of enrollment and those high-risk groups. Were they more likely to be enrolled in the latter portion of the trial and therefore have less time accumulated and at risk? Do you have data about how many people have been followed for 2 months versus 4 months, et cetera, in a different time in risk strata? I'd be curious if there's a difference in terms of the outcomes we're seeing.
Yeah, so we have all the individuals 18 and older were part of our study right from the get-go, as well as all of the risk groups. The only individuals or participants that were invited later as part of the study were the younger age group, the 12 to 15-year-olds, the 16 to 18-year-olds, and individuals that had stable underlying infectious disease conditions such as stable HIV, hepatitis B, or hepatitis C.
Okay, thank you. Dr. Benacloche.
Thank you. My question is, how sure are you that you need the second dose, the booster dose? And the reason I'm asking is that clearly in the New England Journal of Medicine paper, you can see that the responses are much better after the second dose. But in the graph where you show the two curves separating, they separate after 14 days, and the curve remains flat, so you cannot see any effect of the second dose on the curve. And I wonder if there's a possibility that one dose is enough. And if it is not, how do you know it's not?
So we are obviously interested to evaluate in our vaccine study the persistence of protection over time. And so the data that we have so far, and if I could have up the Kaplan-Meier curve, not the insert, but the whole curve, please, if somebody could have that slide up. What you see on that slide, that so far we have, slide 3 up, please. So you see that so far we were able to observe high degree of efficacy for up to 105 days or so. And I think it's really important that we do understand what the persistence is of our protection. So we cannot conclude to say that after a single dose we would see a similar persistence of infection. So therefore, and we also have seen that protection was most mature and was highest after 2 doses were given. So therefore, while we won't have the data to look at 1 dose versus 2 doses, but since we started the study with 2 doses, we of course cannot predict how a single dose would affect vaccine persistence or protection of persistence, which is of course very important in this setting.
Okay, I'm going to, the last question that we're going to have in this session, and we'll try to pick this up when we have more time in the discussion later on, is Dr. Hildreth. So Dr. Hildreth, the floor is yours.
Dr. Hildreth, make sure you don't have your own phone muted. You are unmuted in the system.
I'm sorry, I did not have a question. My question was asked and answered already.
Okay. Well, in that case, we'll move on, at least temporarily. Please stay available so we can return to some of these questions in the first part of our discussion. Now I'd like to call on Dr. Wolsheim from FDA, who is going to give us FDA's presentation of the data and also describe the questions we are going to address in the discussion. Dr. Wolsheim.
Thank you so much, Dr. Monto, and good afternoon, everyone. I'll go ahead and present our FDA review of the clinical data submitted with the Pfizer-BioNTech COVID-19 vaccine emergency authorization request. All right, so a brief outline here. Just to walk you through what we're going to present today, I'll start with a brief introduction of the product and an overview of the clinical development program, followed by our efficacy and safety data analyses, then review the pharmacovigilance plan and plans for future and ongoing studies, and finally, ending with our benefit risk assessment to help prepare the advisory committee to discuss and vote on the topics presented earlier today by Dr. Fink. So first, we will begin with a brief introduction, as Pfizer just walked us through this. The vaccine is based on the SARS-CoV-2 spike glycoprotein S antigen encoded by RNA, and it is formulated in lipid nanoparticles. It's administered by 2 intramuscular injections 21 days apart. The proposed indication and usage under the EUA is for active immunization for the prevention of COVID-19 caused by SARS-CoV-2 in individuals 16 years of age and older. And moving on to a brief overview of their clinical development program to date. There are 2 ongoing studies of the vaccine candidate. Study BNT16201 is an ongoing phase 1 dose-finding safety and immunogenicity study in adults 18 to 15 years of age. And study C459101 is an ongoing phase 1-2-3 randomized placebo-controlled observer-blinded study in individuals 12 years of age and older. I'll note now, as Pfizer also pointed out, that their phase 1 studies did begin at approximately the same time earlier this year, BNT16201 in Germany and C459101 in the U.S. The overlap of vaccine candidates and dose levels in the 2 studies allowed for safety and immunogenicity from each study to provide data and support in real time to change vaccine candidates and dose levels to be evaluated in a larger study that began in the U.S. So looking at study BNT16201, this study evaluated 60 participants who received the vaccine candidate BNT162b2 at the various dose levels listed in the table. There are 12 participants per dosage cohort and no placebo group. The safety results showed a similar safety profile for the vaccine candidate between both phase 1 studies. There are no serious adverse events reported in the safety database for BNT162b2. And moving on to the immunogenicity results. These results were obtained 7 days after dose 2 and showed that 2 doses of the vaccine candidate induced SARS-CoV-2 neutralization antibodies with geometric mean titers comparable to or higher than GMTs from a human convalescent serum panel from recovered COVID-19 patients. The GMTs were highest with the 30 microgram dose. 2 doses of BNT162 also induced S1 binding immune globulin G antibodies and a T helper 1 skewed secretion of interferon gamma and or IL-2. The immunogenicity data demonstrated the prospect of benefit to support enrollment of larger numbers of subjects. and thus the data from study BNT 16201 helped support the final vaccine candidate, selected dose, and vaccination schedule that was determined in tandem with the phase 1 data from study C4591001. We'll move on to that study design next. And initially the study was designed to include approximately 30,000 adults 18 to 85 years of age, and this study population was expanded after the study was already underway to later include participants down to 12 years of age and those with stable chronic medical conditions and infections such as HIV, hepatitis B, or C. All participants were planned to receive 2 doses of the vaccine or placebo 21 days apart. The phase 1 portion of this study was designed for dose finding and vaccine candidate selection based on evaluations of safety and immunogenicity. 90 participants were randomized 4-to-1 to receive vaccine or placebo in cohorts of 15 per dose level, separated into 2 age groups of participants, 18 to 55 years of age and 65 to 85 years of age. The dosing began at the lower dose levels in participants in the younger age group, and the dose was escalated only after safety review, and the older age group was vaccinated with the same vaccine dose and candidate following a satisfactory safety review of the same dose in the younger age group. The numbers of participants who received the various dose levels of the 2 vaccine candidates evaluated are displayed there. These phase 1 results, in combination with those from the study BNT 16201, determine the final vaccine candidate to move forward into phase 2 and 3. All right, let me go one back here. And the study design for phase 2 and 3 included a randomization of 1-to-1 for participants to receive either the vaccine candidate or placebo and to evaluate the safety and the efficacy of the vaccine candidate. The first 360 participants enrolled were specified as an expanded cohort to further evaluate safety and immunogenicity, and any COVID-19 cases from these participants also contributed to the overall phase 3 efficacy population. Enrollment was stratified by age as displayed, with a goal of 40% in the oldest age group, representing a population at risk for COVID-19. The total number of participants 6 years of age and older that were randomized in the phase 2 and 3 as of November 14th was 43,551. And here is a graphic to show the overall study design as planned, with the point in time for the data cutoff identified in yellow, for which we have the data to present to you today. Participants either received vaccine or placebo 21 days apart, and surveillance for potential COVID-19 symptoms began after the first dose. The presence of any of these symptoms would trigger an illness visit for collection of the details about the symptoms and collection of a nasal swab for PCR testing. Additionally, participants had blood drawn prior to dose 1, as well as nasal swabs prior to each dose for SARS-CoV-2 PCR testing to evaluate for the evidence of baseline infection. Participants were vaccinated and followed immediately after each vaccination for 30 minutes for any acute reactions, and then also followed for unsolicited adverse events for 1 month after their last dose, and for serious adverse events for 6 months after their last dose. A subset of participants were designated as the reactogenicity subset and reported daily solicited adverse reactions for 7 days following each dose. All participants are planned to have 2 years of follow-up for possibly related serious adverse events or deaths as well. Moving on to the case definitions here. On the right-hand side of the slide is the case definition for the primary endpoint, which included at least one of the listed symptoms and a positive SARS-CoV-2 PCR test obtained within 4 days of the symptomatic period. And on the right side of the screen is the case definition for one of the secondary efficacy endpoints, which is severe COVID-19 disease. This case definition was met for confirmed COVID-19 cases as shown on the right, who also had any one of the listed more severe criteria, based on vital signs, the need for medical intervention for respiratory failure, shock, intensive care unit admission, or death. And moving on to the primary efficacy endpoints, The primary endpoint definition was using the case definition from this previous slide, confirmed COVID-19 cases that occurred at least 7 days after dose 2 were counted towards the primary endpoint. And the first primary efficacy endpoint was described in terms of COVID-19 incidence per 1,000 years of follow-up in those without serological or virological evidence of past SARS-CoV-2 infection before or during the vaccine regimen. Evidence of past SARS-CoV-2 infection was based on the results from the baseline blood draw to evaluate for the presence of SARS-CoV-2 antibodies prior to dose 2 and the results from each PCR test done prior to each dose, all of which needed to be negative results for participants to be included in the first primary endpoint. The second primary endpoint, efficacy endpoint, was described in the same terms, but in participants with and without evidence of prior infection. The secondary efficacy endpoints are listed here based on different case definitions and time cutoff for counting those cases. The first endpoint listed is for confirmed COVID-19 cases that occurred at least 14 days after dose 2 in those without and with and without evidence of past infection. The second endpoint is confirmed severe COVID-19 cases that occurred at least 7 or 14 days after dose 2 in those with and without evidence of past infection. The final endpoint is confirmed COVID-19 based on the CDC case definition occurring at the same 2 time points in 2 participant populations. And now the next slide will review the statistical considerations for the primary endpoint. The vaccine efficacy was evaluated in participants without evidence of prior infection before 7 days following the second dose in the evaluable efficacy population, which I will define for you on the next slide. And vaccine efficacy was defined in terms of the illness rate ratio, or IRR, where the IRR is calculated as the ratio of the first confirmed COVID-19 illness rate in the vaccine group to the corresponding illness rate in the placebo group. For the interim and final efficacy analyses, the success criterion was met if the lower bound of the vaccine efficacy was greater than 30%, based on the numbers listed for the interim and final analyses above. Interim analyses were planned after the accrual of at least 32, 62, 92, and 120 cases, but in reality, the first interim analysis was conducted upon accrual of 94 cases on November 4th, and it met the success criterion outlined here. The final analysis was planned after accrual of at least 164 cases, and in reality it was conducted upon accrual of 170 cases on November 14th. And now to define for you the analysis populations that we'll show from our data analyses today. The populations and the numbers of the participants who are included in each population are presented here. The all available efficacy populations included all phase 2 and 3 randomized participants who received at least 1 or 2 study vaccinations, and the evaluable... Hello? Okay. The evaluable efficacy population was a subset of this group for participants who received all vaccines within the predefined time window without important protocol deviations and with follow-up through November 14th. The evaluable efficacy population was used for the primary efficacy population. In terms of the safety populations analyzed, the all-enrolled population included all enrolled participants regardless of the duration of follow-up who received at least 1 study vaccination, and the safety population was a subset of this group for participants who received at least 1 study vaccination and were enrolled through October 9th with follow-up through November 14th. So to review the duration of follow-up for the phase 2 and 3 participants in terms of our guidance for the EUA for the COVID-19 vaccine. For this study, I think it's important to note that the sponsor submitted protocol revisions while the study was underway to increase the sample size and to expand the study population. So there was a late enrollment for participants in the adolescent age group, and those over 85 years of age, and those with stable chronic infections such as HIV, hepatitis B or C. So these specific study populations do have less follow-up than the general study population. So for the evaluable efficacy population, the participants included in the interim analysis conducted on November 4th had a follow-up duration of less than 2 months, and the interim analysis was successful with a vaccine efficacy of 95.5% after the accrual of 94 cases. The participants included in the final analysis conducted on November 14th also had a follow-up duration of just less than 2 months. However, this time point included all of the participants included in the interim analysis, and the duration of follow-up for the participants included in the interim analysis at the time of the final analysis is 2 months. We focused our review, however, on the data from the final efficacy data cutoff, which includes those from the interim analysis because the results of the final analysis were consistent with those from the interim analysis, and they provided more robust data from a greater number of participants. The Phase 2-3 safety population includes only Phase 2-3 participants enrolled by October 9th with a follow-up through November 14th, and the median follow-up duration is 2 months. The all-enrolled population includes all enrolled participants regardless of the duration of follow-up. And the median duration of follow-up for this group is less than 2 months, but additional safety analyses from this larger database of all enrolled participants was also reviewed to evaluate for differences compared with the smaller Phase 2-3 safety population. And this is just a duplicate slide. So moving on to one additional consideration that we wanted to point out was implemented for vaccine-induced enhanced respiratory disease. There's oversight from the Data Monitoring Committee from day one, and an unblinded team supporting the DMC review cases of severe COVID-19 at least weekly to evaluate the case splits of the number of severe cases in each treatment group in terms of the study design alert criteria and stopping rules that are outlined in this slide. Neither the alert trigger nor the study stopping criteria were met to suggest that vaccine administration was associated with more severe COVID-19 disease to date. So now I will go ahead and move on to our efficacy data analyses. And the first slide here is showing the demographics of our efficacy population by demographic subgroups such as sex, age, race, ethnicity, and the presence of comorbidities. For age, you can see that the elderly population is represented with approximately 20% of the population being 65 years of age and older. For race, approximately 10% of participants identified themselves as Black or African American, and there are smaller numbers of participants of American Indian, Alaska Native, Native Hawaiian, Pacific Islander, and multiracial backgrounds. In terms of ethnicity, approximately 25% of the population identified themselves as Hispanic or Latino, and approximately 45% of the population had medical comorbidities. Specifically, 35% of the population was obese. You can also see here that there are 153 total adolescents, 16 and 17 years of age, that were included in the efficacy population. The demographics are similar for the safety population. So moving on to the disposition of the efficacy population, to identify the reasons for exclusion from the evaluable efficacy population for analysis of our primary endpoints. Starting at the top of the slide, from the total number of randomized participants, and moving down the table, you will see how many subjects received each dose and were included because they did not have evidence of prior infection, in addition to how many subjects were excluded with the reasons for exclusion listed towards the bottom of the slide. Most of the exclusions were because participants did not receive both vaccinations within the predefined windows, and the number of participants affected was balanced between treatment groups. There were more protocol deviations in the vaccine group than the placebo group, and most of them were medication errors, such as product storage errors, incorrect dosage administration, or the wrong products being administered. And the next slide here shows the results of the primary efficacy analysis. with a total of 170 cases, 8 in the vaccine group and 162 in the placebo group, giving a vaccine efficacy of 95% for the prevention of COVID-19 from 7 days after dose 2. And the criterion for success was met. The pre-specified age stratifications are also shown here with similar results for vaccine efficacy. On this slide, the first row shows the overall second primary efficacy endpoint, which is COVID-19 cases at least 7 days after dose 2 in subjects with and without prior infection. As most people who will receive the vaccine most likely will not know their baseline infection status. The vaccine efficacy was similar to the primary endpoint and had 9 cases in the vaccine group and 169 cases in the placebo group, giving a vaccine efficacy of 94.6%. Subgroup analyses of the second primary endpoint are also shown here by age, obesity as a comorbidity, and by sex. The vaccine efficacy was consistently high across different subgroup analyses, with limitations on the adolescent age group because of small numbers of cases and participants as compared to the adult population. The vaccine point estimates were uniformly high across the subgroups examined, with the exception of participants identifying as multiracial and participants with evidence of prior SARS-CoV-2 infection at enrollment, for which too few COVID-19 cases occurred to interpret efficacy data for these subgroups. So a continuation of the subgroup analyses for the second primary endpoint are shown here. I think I just described that with the prior slide. So the vaccine efficacy point estimates were uniformly high, so you can see the numbers here on this slide. And moving on to one additional subgroup analysis of the vaccine efficacy by comorbidity, to show that the vaccine efficacy point estimates, again, were uniformly high across subgroups examined that increased the risk of COVID-19, such as chronic pulmonary disease, diabetes, obesity, and hypertension. The next slide here is to show that the secondary efficacy analyses had similar case splits, and the vaccine efficacy results were consistent with the primary efficacy endpoint for these secondary efficacy endpoints as well, based on the case definitions, with cases being counted at 14 days following dose 2, as well as using the CDC case definition. The other secondary efficacy analysis to discuss is severe COVID-19 cases, and this table breaks down the numbers of severe cases that occurred in the all-available efficacy population at each range of time points shown following dose 1. So in the all-available efficacy population, 10 participants had severe COVID-19 disease after dose 1, 1 subject who received the vaccine, and 2 subjects who received placebo. The vaccine efficacy was 88.9%, but with a wide 95% confidence interval that goes down to 20%. Based on review of the case narratives overall in the all-available efficacy population, 2 placebo recipients met severe criteria based on vital signs and no other criteria. 6 placebo recipients who had severe COVID-19 disease were hospitalized, 3 of which were admitted to an intensive care unit, and 7 placebo recipients had at least 1 risk factor for severe disease. In the bottom row of this table, you see the case split for the primary endpoint evaluation at least 7 days after dose 2, in which 4 placebo recipients and 1 vaccine recipient had severe COVID-19 disease. The vaccine recipient who had severe COVID-19 disease met the severe case definition because of oxygen saturations at the time of the COVID-19 illness visit, being less than 93% on room air. The subject was not hospitalized, did not seek further medical care, and did not have risk factors for severe disease. The 4 placebo recipients who had severe COVID-19 disease met the severe case definition for the following reasons. One subject had oxygen saturation of 92% on room air, and one had a heart rate of 125 without other severe criteria. One subject was hospitalized for non-invasive positive pressure ventilation with bilateral pneumonia. One subject had an oxygen saturation of 92% and ICU admission for heart block. 2 of these placebo recipients with severe disease also had obesity as a risk factor, while the other 2 participants did not have any risk factors for severe disease. The total number of severe cases is small, which does limit the overall conclusions that can be drawn. However, the case split does suggest protection from severe COVID-19 disease. We'll move on to one post-hoc analysis that we ran on the timing of COVID-19 cases following dose 1 in the all-available efficacy population. The timing of cases is presented similarly to the prior slide for severe cases. Vaccine efficacy in participants in the all-available efficacy population was similar to results in the evaluable efficacy population. And the vaccine efficacy for prevention of COVID-19 disease after dose 1 is 82%. Based on the number of cases accumulated after dose 1 and before dose 2, you can see that there does seem to be some protection against COVID-19 disease following one dose. However, these data do not provide information about longer-term protection beyond 21 days after a single dose, because the vast majority of subjects received dose 2. And our next slide here shows one additional post-hoc analysis of vaccine efficacy based on baseline SARS-CoV-2 status in the dose 1 all-available efficacy population. Only 3% of participants had evidence of prior infection at study enrollment, and additional analyses showed that very few COVID-19 cases occurred in these participants over the course of the entire study. 10 in the vaccine group and 9 in the placebo group. Only 1 of the 10 cases in the vaccine group occurred 7 days or more after completion of the 2-dose vaccine regimen. So some participants with prior evidence of infection also had confirmed COVID-19 during the study conduct. These data do suggest that previously infected individuals can be at risk of COVID-19 reinfection. The small numbers of participants with baseline positive status limits the interpretation of the efficacy at this time point. Moving on, we will now focus our attention on the safety data. And here's a quick reminder from the graphic about the general study design. And first we'll review the reactogenicity results collected daily for the first 7 days following each dose in the reactogenicity subset. Dose 1 reactogenicity is shown here on this first slide, and they're separated by the predefined age stratification. For both age groups, injection site pain was the most frequent solicited local adverse reaction. After dose 2, the younger age group reported any pain more frequently than the older group, and pain was characterized as moderate, with a similar pattern observed after dose 1. Injection site redness and swelling after each dose were generally similar for both age groups. For each age group in the reactogenicity subset, the younger subjects, and overall, the median onset of local reactions in the vaccine group was from the day of vaccination to 2 days after the dose, and lasted for a median duration between 1 and 2 doses, 1 and 2 days. Local reactogenicity following dose 2 is shown here, with consistent reports of pain, redness, and swelling. And analyses by various demographic subgroups showed consistent results. The next slide here shows, my slides are a little behind, I'm sorry. So here are solicited adverse events within 7 days following dose 1. I'm caught up, I apologize for that. These are, let me catch up here. These are reports of joint pain, diarrhea, and vomiting, which were reported pretty equally between the age groups. And following dose 2, the systemic reactogenicity is shown here. The frequency and severity of systemic AEs was higher after dose 1 than in dose 2, than dose 1, except for vomiting and diarrhea, which is generally similar regardless of dose. And again, analyses by various demographic subgroups showed consistent results. This slide is a continuation of the dose 2 solicited systemic adverse reactions within 7 days, showing the numbers for joint pain, diarrhea, and vomiting. Here are the numbers for our adolescents, 16 and 17 years of age. It's important to note that for the majority of adolescents, 16 and 17 years of age, enrolled by October 9th, local reactions and systemic AE data were collected as unsolicited AEs with the preferred terms shown here, rather than from pre-specified prompts for reactions, which were solicited daily for 7 days for each vaccination, as was done for the adult population just shown. So the same details are not available for reactogenicity in 16 and 17-year-olds as compared to the adults, and instead